Hello, hello, check, hello, test, check, hello, check, hello, hello, check, hello, check, hello, check, hello, hello, check, hello, check, hello, check, hello, check, hello, check, hello, hello, check, hello, test, hello, test, hello,
हेलो हेलो चेक कराए थे
Hello. Good morning, all. Respected faculty members, invited speakers, delegates, ladies and gentlemen, a very warm welcome to the 71st Annual National Conference of Physiology and Pharmacology with the interesting theme that is Physiologist and Pharmacologist for Fit and Healthy India. This Association of Physiologists and Pharmacologists of India was established in 1957 with the vision of promoting research, education, and collaboration in the field of physiology and pharmacology across the country. The Belgium branch of APPI was established in 1992 and has been one of the most vibrant and active local chapters. We have also been honored with the Best Branch Award for 7 times and consecutively for 5 times. That shows excellencies of our association. It is indeed a moment of pride that the Department of Physiology and Pharmacology, APPI Local Belgium Branch, is hosting the event at KLE Academy of Higher Education and Research. The last Epicon held in Belgium was in 1995, and after 3 decades, we once again have the honor and privilege of welcoming this prestigious gathering to our city. Kunda Nagri, Nama Belgami. With that note, let us commence today's session with great enthusiasm and a spirit of learning, sharing, and collaboration.
For this very first session on topic fitness of germ cell and stem cell, a new era in regenerative medicine, I request our chairperson, Dr. Anand Bhaskar sir, Professor Christian Medical College Vellore, and Dr. Sachin Rathod sir, Associate Professor AIMS Nagpur, to please come on the stage and chair the session. Now I request our chairpersons to introduce the esteemed invited speaker, Dr. G. Taru Sharma.
Good morning to everyone. It's my privilege to introduce our esteemed invited speaker, Dr. G. Taru Sharma, Director of the National Institute of Animal Biotechnology, an autonomous institute under the Department of Biotechnology, Ministry of Science and Technology, Government of India. Dr. Taru Sharma is a distinguished scientist whose pioneering work in the germ cell and stem cell biology has greatly advanced our understanding and the regenerative medicine. Her research has opened new pathways for cellular and molecular innovation in health and biotechnology. Today, Today, she will speak on the fascinating topic that fitness of germ cell and stem cell, a new era in regenerative medicine. We are honored to have her with us, Appicon 2025, and we eagerly look forward to her enlightening the talk. Please join me in warmly welcoming Dr. G. Taru Sharma, sir.
Okay, we will move to the next session. The topic is role of physiologist in Fit India by Dr. Chinmay Shah. I request the chairpersons to introduce the speaker and moderator.
Good morning, everyone, again. It's my pleasure to introduce our distinguished invited speaker, Dr. Chinmay Shah, sir, Dean and Professor of Physiology at Government Medical College, Bhavnagar. Dr. Shah, sir, is a highly respected academician, teacher, and researcher who has contributed immensely to the field of physiology education and applied health promotion. Sir has a keen interest in medical education technology and has been actively involved in enhancing the innovative teachings and assessment strategy for the physiology learners. His research also focus on the scientific writing and publication skills, promoting academic integrity and evidence-based communication among the medical educators and students. He has been a strong advocate for translating physiological knowledge into community well-being, particularly through the national health movements like Fit India. Today, he will be sharing his thoughts on a very relevant and inspiring topic, role of physiologists in Fit India. This talk will help us and appreciate how physiologists can play a pivotal role in promoting the physical fitness, preventing health and lifestyle awareness across all age group, integrating science, education, and public health outreach. Let us all join in extending a warm welcome to the Dr. Chinmay Shah, sir. Sir, we look forward to your insights.
Uh, Very good morning to everyone. And first of all, thanks to organizer and thanks to the people who are here in this early morning to uh, share their views with me. Okay. So, okay. Let me start my session. So I'm happy that I'm the opening batsman of the session. Favorite slide all over that all the contents are from various sources. Information is only for educational purpose and there is no conflict of interest. I acknowledge the theme as well as KLE, KAHER, APPI, and the organizing committee of Appicon for inviting me and giving me opportunity to share my views. So, all the sessions start with the objective. So in the delegate group, I have shared the link. So I request all of you to why you are here. So can we have a few seconds? I can say 10 seconds, 20 seconds. You can write one objective for which you are here. I have shared the link in the our delegate group. You can take 20 seconds and share. I got one response, that is knowledge, for knowledge. Okay. For update. See, in today's era, okay, regarding latest things in Fit India. Okay, great. So moving forward, let me share objective of session, what we are going to discuss today. So we are trying to basically discuss what is Fit India, what is the role of physiologist, and how a physiologist can play a role in public as well as private sector. Okay, and lastly, what are the challenges and opportunity? So as you all are aware that in August 2019, to convert our unfit citizen to fit citizen, uh, the Department of Youth has started this movement and launched Fit India. And the Fit India movement is implemented effectively only with the help of physiologists and will continue to grow with the help of physiologists that we will see in coming slides. So basically, what was the purpose of Fit India movement by Government of India? Starting with the promoting fitness, encouraging fitness in a way of easy and fun way. And there are a lot many things they have provided free. So there is no any expense to the people who are trying to get themselves fit. Then expanding the awareness. So starting from school, colleges, different clubs, they want to increase awareness by Fit India. Reaching diverse community, as I said, school, colleges, villages, and panchayats. And lastly, fostering indigenous sports, supporting and promoting traditional Indian sports. So these are the basic objective why Fit India movement was started. So when it was started, there is an abstract health goal which can be a vague and have an intangible objective. With the help of scientific approach, by adding a knowledge and methodological things, we are converting that into the tangible and sustainable actions. And we want to develop a population level health by improving health for everyone. And here a role of physiologist comes. So physiologist is a bridge who is going to convert abstract health goal into actionable behavior. So basically, in a three different way, they have envisaged the role of physiologist, because the upcoming branch of physiology, popular known as exercise physiology, and sometime it is interchangeably used with the sports physiology, that will help to study different changes happening during the exercise and sport, and help us to understand how the body adapt. Where after involvement of physiologist, we are going to collect the data, we are analyzing the data, and based on that, we are going to guide and coordination coach and sports psychologist. But in the team, what is the role of physiologist? The our main role is physiological testing, performance analysis, exercise program design, health management for chronic conditions, with the primary focus on systemic physiological adaptation and overall health advancement. And as CBME said, the skills which we require, physiologists require, are exercise testing, data analysis, metabolic assessment, and program development based on scientific principle. But our work will not be complete without the team of the physiotherapist, strength and conditioning coach, as well as sports psychologist. Now let's focus a little bit on exercise physiology or sports physiology, which is a key for a fit India. If we look at the Indian context, it is a scientific branch or a scientific study of how the body responded to the physical activity, especially focusing on enhancing athletic performance, promoting health, and preventing chronic disease through evidence-based strategy. And what we are going to do is, we are starting with the understanding our human body, what are the acute response to the exercise, chronic response to the exercise. We are going to apply this scientific principle using a research and guided practice. And ultimately, we are going to come with the, we can say a menu, we can say a guideline, which optimize the training, enhance performance, and accelerate the recovery in case of the damage. So let's look at the few government-led initiative where physiologists are playing a role. So as we already know, Fit India movement, launched by the government with aims to promote healthy lifestyle. Now there are fitness protocol. If you look at the website of Fit India, so there the physiologists have played a pivotal role by creating and helping a campaign uh, how the direct application of different exercise deal with the different age group. This makes physiology not a peripheral science, but a core enabling discipline for the entire campaign. Its principles provide the scientific rigor to move from abstract goal to the measurable, effective, smart goals, as we have discussed previously. Now, there are three important parts where physiologists have played a role. One is the goals, a scientific framework, which includes the taste and activities targeting flexibility, strength, endurance, and cardiovascular fitness. And because of these, the official government endorsement to the activity done by the physiologist, it demonstrates a specific evidence-based recommendation, and which is catered to distinct population and segment needs. And ultimately, because of the development or involvement of physiologists, there is a protocol validity and impact based on the different guideline provided by us. We know Dr. K.K. Deepak, who is the chairman of expert committee for the development of fitness protocol, and he has played a pivotal role. And he says that physical fitness is a necessity to enable us to perform and carry out all our activity of life. Human well-being is always desirable in all age group. And Kavi Kalidas said, Sarir Madam Khalu Dharma Sadhanam. It means that the body is the foremost medium to perform the duties. Now, as we have seen in a government sector, there is integration by Sports Authority of India. Now, to create an awareness, Sports Authority of India has created National Center of Excellence and has decided a sports science support. And in this support, there are different 6 pillars, and one of the pillar is physiology. The government is going to spend around 80 crore to set up a various sports science, and the explicit inclusion of physiology as funded pillar signals fundamental shift away from traditional ad hoc coaching method to the integrated science-based approach to athletic training. The ongoing hiring of high performance analysts in various sports science roles, including physiologists, underscores this institutional commitment. And so by this, even now we are getting an uh, recognition as well as endorsement. If we look at the case studies of National Center of Excellence, there are around 23 disciplines, and out of which the role of physiologist and other scientific experts are there to evaluate, support the performance of young athletes. So again, we are getting our role as a physiologist. The thing what our the case says that we need to start integration of physiology at the early stage, starting from the policy making to the implementation. Even if we go more ahead with the other disciplines, Sports Authority of India as well as IIT Delhi has done a partnership with the help of physiologists to bring bridging the theory into the applications. And now we are focusing on the technology, how we can get more and more data. And ultimately, we are going from ad hoc to data-driven paradigm. This was regarding government. Now, if you look at the private sector, which is more in technology and innovation, we have a two important thing, FitPass and Fitter. Now, what these two people are doing and how the physiologists are playing a role here. If we look at these two institutions, it says how physiological principles are being commercialized and scaled. Now, Fitter model emphasize there is a tailored plan for individual people. And so they focus on building a sustainable habits, and they also help in injury rehabilitation. And this integration of both building a sustainable habit as well as injury rehabilitation with a single platform mirrors the multidisciplinary approach seen in SAI's National Center of Excellence. Now, if we go into the details of FitPass, they have two different things, Fit Coach and Fit Fist. So what this Fit Coach is doing? So in Fit Coach, the physiologists are providing a theory and principles. So initially, when we are at a distinct level, we are only catering to a small group of people. Now, with the help of AI, we are validating the AI, and now we are expanding our vision and our horizon to help society at large. If we look at the role of physiologists in FitPass, we are providing educational content, we are ensuring the safety, we are creating workout progress, we are enabling personalization, and ultimately setting progression logic. So here comes the role of physiologist. Again, same way, if we look at the fit fist, our physiologist has to play a role in a form of connecting exercise and nutrition, creating a content for health and wellness, creating a user assessment tools, advising on the dietary science, data analysis and health challenge. So we need to introspect and see that these are the field where we now need to deep dive. Now, if you compare government and private, in a single word, I can say that government is focusing on more or awareness, while private institutions are focusing on individualized approach. So now we can play either role. Either if we are on the government side, we can go and work on the large scale. And now there are lot many clinics, lot many institutions are opening up only for sports and exercise, where we can cater need as per the individual requirement. If we broadly divide them, so the primary goal of Fit India is, or SAI is, mass scale behavior change, fostering a culture of health and elite athlete performance, while a Fitter and FitPass provide personalized, on-demand, and technology-driven fitness and wellness service to consumer for the profit. Now, as we have already discussed, both have a role of physiologist in expanding the horizon, and the key challenge which they both are addressing are for government sector, they are addressing widespread health literacy, poor infrastructure, and a lack of access to basic fitness resources, while private sector is addressing a scalability of personalized service, user engagement, and providing on-demand affordable solution to large population. Now, if you want to grow further, there are certain challenges. There are demand and recognition of physiologists in the field of Fit India. So these are the common problems which usually often when we discuss systemic corruption, cultural bias, uh, inadequate infrastructure, and elite career pressure. So if we trying to address these, we are definitely going to make an impact in the field of Fit India. Now, there are logical and technological gaps. We need to do more research into sports gear industry. For example, issue like logistic insufficiency, high cost, low technology. So our honorable prime minister is suggesting make in India and promoting startups So we need to go in it, and whatever is available outside India, try to develop inside the India. And I think on my experience, I have seen that the cost will reduce to 10% to 20%. So that we have to do, and we need to talk with each other. Right now, sometimes anatomy, physiology, biochemistry are not talking, but we need to go beyond our branch, and we need to talk with the IITs. We need to talk with the other industries, and we have to take them into consideration in promoting health. We also need to do the research interdisciplinary. So again, I will quote Dr. K. Deepak. Right now, he's a professor at IIT Delhi. So now he is guiding IIT people for different, based on different physiological principles. So it's our again role, and as a society, as our association, we need to promote interdisciplinary knowledge. Now demand is increasing, as we have seen that in government, SAIs, then National Center of Excellence, then FitPass and FitCoach, our need is increasing. So I can see few young minds. So now it's our role, our senior people's role is to inculcate this thing in our young budding resident doctors, that this is the field where you can grow. And it is not like a technician. See, in other places, lot many clinical applications we have started in physiology, but sometime at the end of the day, we feel that are we technician or are we physiologist? But this is the way where you can coach, you can guide, you can prescribe exercise as well as nutrition. Uh, so there are a lot many IPL, ISL, Pro Kabaddi League, everything is coming up in a big way. So we need to be a part of all these different private and government funded activity, and we need to involve more. If we look at the Reliance, they have started with the mobile. Kar lo Duniya Mutti Mein was only 500 rupees, 500. And now they have started in a big scale. So we need to go inside in any way in all these fields, and then we need to grow. Uh, Again, coming to the role of professional bodies, as I'm thankful to the organizing committee for taking up this particular topic as a theme of conference, that as a professional body, if we will start developing a certification or something like that, there is one of the association which is coming up nicely is Indian Society of Sports and Exercise Medicine. But we need to reach to the level of American College of Sports Medicine. So I suggest and I request the leaders of APPI to look into this matter, and we can start certification exam, develop a module, develop a curriculum for a sports, and we can start certification. Now, what is the next? If we are going up to this level, the next will be a data-driven approach. We need to start more and more study on HRV, then resting heart rate. We need to involve the effect of all these different sports exercise on autonomic nervous system. And if we start producing our impact in the form of research, दुनिया सबूत मांगती है, it is a famous dialogue of Hindi movie. So if we will start developing our own data, we have a data, but Indian data is lacking, so we need to go for Indian data. And once we are there with our data, we can mark a bang. And lastly, recommendation and strategy, the blueprint. So first is protocol standardization. We have a protocol for a layman, we have a protocol for the society, but now as an association, we can come forward and develop a protocol for a, I can say service, for a professional service. As a medical college people, it is our role to integrate into curriculum. CBME have started 2 hours per week for a sports, but it is just for a sport, but not a curriculum integration. So we need to do that, and we need to involve in clinical applications of all the physiological principles. We need to strengthen the public and private sectors, because public people are working on their own, private are working on their own. If they come together, we can have a tailored solution as well as public awareness. As I already said, professional development and certification is required, and API can take it forward. Lastly, we need to create a national-wide public awareness campaign, highlight a fulfilling careers, because ultimately our what resident look into career. I'm going to get a job? Am I going to get money out of it? If we can showcase them this, then people will go towards it. And inclusive movement for all. There are few policy recommendations which we can share. It is a financial wellness incentive. Now, what is this? That if any institution, if any company is spending some money for any activity which promote fitness, government can give them financial incentive. we need to create more sports awareness campaign as well as spend something on sports infrastructure investment. So to conclude my uh, talk, the Fit India movement has successfully established a national dialogue on health and fitness, but in long term, only it we all get benefited if the physiologists get part and parcel of it. And the physiology is no longer a peripheral figure, but a central to this movement. And contribution span from designing population-wide health guideline to optimizing a performance of elite athletes is a role of physiology. Thank you. Thank you very much.
Thank you, sir. Thank you very much, sir, for motivating thought, provoking regarding the sports as a part of the curriculum again. Your session beautifully emphasized how physiologists can move beyond the classroom and in laboratories to actively contribute to community fitness, health awareness, and national wellness initiative. Fit रहेगा India तो ही आगे बढ़ेगा India. Thank you very much, sir. On behalf of Appicon 2025 and all participants, I sincerely thank you for sharing your vision and enthusiasm with us today. Thank you, sir.
Thank you. Thank you very much.
Thank you, sir. Now I request the chairpersons to present the certificate and a memento to our resource person. Now we will move on to the next session. The topic is fitness of germ cell and stem cell, a new era in regenerative medicine. I request the chairpersons to introduce the invited speaker, Dr. G. Taru Sharma.
Okay, it's my privilege to introduce our esteemed speaker, Dr. G. Taru Sharma, Director of National Institute of Animal Biotechnology, an autonomous institute under the Department of Biotechnology, Ministry of Science and Technology, Government of India. Dr. Sharma is a distinguished scientist whose pioneering work in germ cell and stem cell biology has greatly advanced our understanding of reproductive and regenerative medicine. Her research has opened new pathways for cellular and molecular innovations in health and biotechnology. Today, she will speak on the fascinating topic, fitness of germ cells and stem cells, a new era in regenerative medicine. We are honored to have her with us at Appicon 2025, and we eagerly look forward to her enlightening talk. So please join me in warmly welcoming Dr. G. Taru Sharma.
I think I'll just put my presentation. Can someone help me out here? Where is it? Very good morning. Thank you for the introduction. There is a small change in my introduction. Yes, when I was invited by her, I was at Hyderabad. I am transitioning to another institute, Indian Veterinary Research Institute. This is for your information, not your fall, but it was this way. So first of all, I really thank Dr. Harpreet. She's not here. I believe very concentrated, few most important people are in the hall. That's how I feel. Very good morning, Professor Shashidhara, and all others. I only know him. Thank you very much for that. And when I was asked to do that, I'm a life member of Appicon, and I'm also founder president of another physiology association, which yesterday we had a meeting over there. So just thinking like which way, I don't work on a sports physiology. I am not into, any of the activities which are directly linking to sports. Then we sat and discussed, and she said we can really do this. And when I say page down, it doesn't move. So sorry for this glitch. No, when I'm putting it enter, it doesn't go. That's the point I'm making. Yes.
Down.
Okay. Yes. Thank you. So, since I work mainly on stem cells and also the female germ cell oocyte, anything and everything to do with large animals. So, fitness and life, so life continues with the germ cells and it gets repaired through the stem cells. So then I decided why not I talk something on a fitness of germ cells and stem cells. Once upon a time, everybody started as embryo. And the oocytes fitness and the fitness of a stem cell when we go for the regenerative therapy. So that's how I just thought it could make sense to the esteemed audience. So when we see the convergence of both, and when I say the germ cell and stem cell convergence over here, surely I'm trying to talk about the pluripotent stem cells. Nonetheless, I'm also making a point that mesenchymal stem cells also, which are of a mesoderm origin, can even that also can be linked with it. So all in vitro gametogenesis or in vivo gametogenesis. And when we move for the therapy part, surely it is that. Well, I think I just thought I can elaborate a little bit over here. The point which I'm trying to make out here is what exactly is an interface? Again, I'm not touching iPSCs here. I'm just trying to talk about either the PGCs or the PSCs, right? Pluripotent or the totipotent stem cells. Moving on to that, before I come and share the data, how exactly we see the fitness of the germ cell, let me just, pinpoint here, what could be the determinants when we think about the germ cell fitnesses? I think the circle in itself is loud to say, but what are the areas wherein we work on? So we work on in this mitochondrial and metabolic health of the female germplasm with an idea that how could you increase the shelf life of the female germplasm, and especially in an animals and large animals where the gestation period remains as much as is for human, that is 9 months for both cattle and buffalo. So what exactly you could do to rescue major number of eggs to that? Number one, we do also work on epigenetic reprogramming and also the developmental competence along with the cellular qualities. So what I'm going to do is I will just focus a little bit on the oocyte data and how exactly we can link it with the mitochondrial health and the metabolism and the stresses. Okay, so with that I move on. That's a picture. Well, these are all eggs of caprine origin. The point we raised, please do note, other than the first one, which I say the excellent egg, none of the other ones have the surrounding cumulus cells. So the questions we raise is that are the oocyte surrounding cells so important that if there is an alteration, it could go for impediment? And mainly since ours is a IVF system, so we were concerned with that. Why did I really show the staining out here? So the answer of this question is, to this particular talk, I could not get all the bunch of data. But answer of this is no, they are not. There is internal machinery besides mitochondria. The development of microtubules and microfilaments make a very important component other than the ROS and the calcium, which I will cover over here. Okay, so we looked at from a different frame and we started focusing on how could we really decode the metabolic regulation which could lead to a better maturation, or rather put the egg towards the maturation and surely the fitness. And so we just looked at the diagram. We all know that the oocytes and early embryos, they are all pyruvate dependent. So everything and anything to be done with the pyruvate. So we just decided with that and focused up to the onto the oxidative phosphorylation pathway. Do note that I'm making a arrow over there with NADH. So naturally NADH, and we thought and we could generate the data. It's a metabolic environment which is very critical. One can always debate why not cumulus cells for sure, but cumulus cells when the egg is growing in vivo and then hormones have to reach over there. So a post-theca externa, nothing reaches directly through circulation. That is how they're important. So high ROS and high calcium, they are the two major causes which really are responsible for depleting the membrane potential of the mitochondria. This is a statement based upon our data and was a data. So what did we do? We collected the best quality eggs and the worst quality eggs. So we say excellent and poor. That's how the school of thought goes on. Sorry. We stain them with the JC1, which is a mitochondrial dye. And you could see over there what is exactly is the signal intensity shown unto that. Keep this, keep this in mind and move on. The important point, and to our surprise, when we stained with the mito tracker dye, the green ones, we understood and look at that picture. The mitochondrial distribution is not at all compromised in the poor eggs. And that's a boom. Like one can really think of getting them into the cycle. And increase ROS, as I was just telling over there, just see the picture, how much difference. So what we made, the first statement based upon a previous data, it's a truth. And then when we have gone further for the calcium levels, again, it's a truth. So we could really corroborate it. And this is the important lead, which we made 2.5 years back. And then we are now building upon it. And that is how the fitness of the germ cell is with us, which leads to longer shelf life, getting more number of oocytes on the plate, and more number of oocytes, or more number of embryos, more number of blastocysts, which are transferable. Let me just move on with that. So clearly, if you could just hang the numbers and say, besides all other things, I would just restrict to this mitochondrial metabolic support. I said it already. And of course, this lifestyle factors, the gentleman before me in the talk was saying. So we know, all of us know leptin and ghrelin, one good, one not so good. So leptin receptor and kisspeptin, a Kindi's neuron, the moment you connect them, uh, lower level of leptin, lower level of Kindi neuron, lower level of reproduction, for sure, everything fits in so well for towards the fitness of either the physical fitness or the reproductive fitness. This becomes much more important. Why? Because we all know this all has been put to the pause in any of the physiological system across the species if the basic needs are not fulfilled. Let's just look at it. What could be the probable threats? I think these are the probable threats. Of course, what I have written, it's hormonal dysregulation, PCOS, and stress, and several other things. It does have a clinical relevance too. The last two lines which I just wrote, using 2 years back references, how interesting. It was actually, I also got educated. If we just take a damaged sperm and it will fertilize the oocyte for sure, because what you need is the mitochondrial thing should be on and it should just go and you should make a 2N. But the blastulation rate and further gestation and the miscarriage risk increases. Whereas if we take a younger oocyte and repair this DNA fragmentation, it will just safeguard the embryo development. So I'm keeping it short because I don't work in an area, but I thought it makes a sense to talk over here. Let's just switch gears. I was told that I've got 25 minutes, so I'll respect that. Let's move to stem cell fitness. So whatever is all keeping the long story written short, it's a busy slide, but I'll keep focused on that what exactly is the ability of their self-renewal, be them embryonic, be them adult. Because anyway, I had to link it with the regenerative therapy. On this picture, people who are aware with the basic physiology, with the basic stem cell biology, understand you got to prove them that they're stem cell by all the ways, trilineage or maybe surface markers and so on and so forth. And one could think of there are enormous possibilities which we do. We use mainly the ones which are written on the right, left-hand side. We use umbilical cord not only to derive the fetal stem cell. The whole idea is because teratoma, they are non-teratogenic cells, but the telomerase activity is higher and they are not embryonic, so there's no teratoma formation. We also work on bone marrow derived stem cells. So this data I shall share today. Linking with the fitness. We also work on secretome a lot. We work on exosomes and also we do a lot of immunomodulation because these are immune compromised cells, and that's how majority of the regenerative medicine is rotating around the the adult stem cells, particularly the mesenchymal ones. So this is the arrangement, and when we talk about this, it surely regenerates, restores, and replaces. Well, one can say that when you're going for that, anyway, physiologically it's happening. But if a doc comes to your clinic, if you can put the owner's agony lesser by 10 days or 2 weeks, that's a help. So this is what we do a lot, but I'm not going to talk about the secretome or exosomes or and everything. I just talk about the fitness of the stem cell. So when we talk about the fitness, that means how do they signal to each other? What are the signaling molecules playing role, and how do they get converted towards a particular lineage? I'll talk about that. Utilizing in the sorry, regenerative therapy. As I told, we also generate lot of scaffolds, but the topic is fitness, so I shall restrict to that. They have homing properties, they are immunomodulating, immune compromised cells, and so on and so forth. So moving on with that, this is what we do. We collect it from the marrow, and once you have the cells, you say that through all the markers that yes, they are the MSCs, and then you move on with that. And once you have it, what exactly they do, they get transdifferentiated to trilineages, and if yes, it's fine. So they get differentiated to adipo, chondro, and osteo. But since we work on two major clinical aspects, one is a surface repair and another is a bone repair. So osteogenic lineage, I chose to speak of the signaling pathway of the osteogenic lineages and what does it do. So few questions which are in front of us. And, this meeting made me think that how well things get fit into the fitness of any of the research work with utilizing the cells, be it a viral cell, a bacterial cell, or a germ cell, or a stem cell. So these are the relevant questions which we have had for ourselves. And to address all these things, what determines the weight so that they remain fit? And if you have the signaling mechanism understood to you, That's the answer of it. Surely the adipogenic, and that's how you know, and the chondrogenic. But as I said, I shall focus on the osteogenic with the beautiful staining, and it does say that your cells are doing pretty well in the trilineage differentiation. So osteogenic lineages, mainly three pathways, BMP, Wnt, and Notch. Of there is a crosstalk. No one can just say just three pathways. There's a crisscross and lot of things to be done. Maybe I can just skip in a half a second, but it's an idea to show that it's osteopontin, osteocalcin, and then how exactly they get differentiated through the process of MSCs, and that's how they help the whole thing. So there are intrinsic factors and the extrinsic factors when we talk about the cells, which they are moving from proliferation till the osteocyte making. And we are concerned with this because we're talking about the osteogenic lineages. So the points I wrote over here makes us important sense. And there is a finer balance. As I said, the pathways are never so clear. So you do have a crisscross, and there is a bit of dwindling. So there's a balance and to happen when it goes from adipose and osteo. So there is a connection between the two. I'll keep the story very simple. So we focused on RunX2 mainly over here, and that story I'm going to narrate fully. So as I say, that BMP, little bit I touch, but first I'll talk about the RunX because we treated RunX as a central dogma, and the SMART story, SMART pathway, whole SMART pathway. Two, we all know, independent and dependent, and how exactly it's put over here. So basically, the RunX2, where it is modulating between the adipo and osteo, what I'm trying to talk about the receptors and its binding with the SMAT proteins. And this you have to work out through the markers in any way, the staining or whatever you do. But BMP controls the expression of the transcription factors, which I have written down over here. And RunX2 via SMAT signaling is the one which is a central dogma. Okay. So I move on, and I think notch pathway, I'll just skip this slide. I'll not skip because I had to talk one important part over here, the gamma secretase. Please remember, because we have to talk about the gamma secretase when we talk about the RunX and connect this with Hess and Hay. Okay. So if we suppress both the pathways, what the signals will do, and will it affect the activity of the RunX2, the transcription factor, and ultimately how the differentiation will go on. So this is a dimorphic model, and that's what I was just saying. So this is a very simple picture till now what I was trying to talk, the BMP to SMAT to RunX, and then eventually the differentiation and Hess and Hay, two genes I just spoke about. So we could elucidated that. These are the cells, and this is how we have just subdivided our groups to understand what exactly is happening. So I'll move on to show and share that it was important for me to show the subgroups so that I can keep people with me when I move on. And this is what we just said. We just said, this is on a canine picture, as I said, once we had done it, and after all this understanding, we have said that what we propose and question, we have said it the same way into our answer. And this is what is a data in a very capsuled way. It's a published work now. 22 we published this one. So what we can just understand over here, the third group, if you remember, which is a gamma secretase, we simply use a shRNA platform over to understand the signaling. So the third one, which is a gamma secretase model over here for osteopontin and osteocalcin, and Hay and Hess picture, we really did it. After this, all the cells, again, we allowed them to go for the osteogenic differentiation. And this is what we said in the end. We said that if you silence the BMP pathway, it would impair the in vitro osteogenic differentiation of the canine origin adult stem cells, MSCs. and it is promoted by Runex. Exactly what I showed in the questions which we raised. That's the work which is a research in veterinary science. And one important thing I must mention, that BMP dependent SMAD4 signaling is the one which is positively regulating the osteogenic differentiation, wherein the notch dependent Haven signaling is not supporting it, it is rather inhibiting. So then we are right now moving with the SMART pathway. You may put a bell when you feel that I'm left with 5 minutes. So now we are moving with the SMAT4 and BMP pathways. And this is the bars which we hang for ourselves. One story I narrated, this one. And then we thought that why not we include the TAS, because TAS molecule is a central molecule which is which it could get diverted towards adipogenic depending on the pathway, else it can remain to osteogenic. So we thought, why not we work with the independent pathway first, based upon this paper, and focus only on osteogenic pathway. So we raise a question to ourselves. It's a very busy slide, but don't worry, I'll make it very simple. Just remember that red colored TAS expression. So by now we understand SMAD-led RUNX through transcription factor RUNX2 and Hersey pathway. Then what exactly is TAS doing? So this is what is for osteogenic, but as I said, the pathway which is a SMAD LOX pathway, which has an adipogenic commitment, do they crosstalk to each other? If you remember 5 slides back, I said there's a delicate balance between adipogenesis and osteogenesis. Do note, I never brought chondrogenic lineage in this discussion. So that's a good news for the researchers, so that you can only focus on adipo if you're working on osteo, not that. And then we moved on with that. So this is a cytosolic picture, SMAD and TAS over there, and this is a nuclear picture. And that's what I was just explaining a while ago. So dependent and independent signaling. As I said, we decided to move on with the other one. So that's a cytosolic SMAD dependent signaling, which is leading to, again, one has to remember the ligand binding would work through dependent pathway. However, there could be an independent signaling which could lead to the regulation of the osteogenic lineage. For the independent, we raise a question, is SMAD4 talking something or getting translocated to nucleus? Or are there some other signaling pathways which are doing that? Question. We have solved it. All the data I may not share, but some data I will share over here. Nice, clear, simple picture. What I explained till now, I'm just saying the same thing. If you do not allow BMPR, then what happens? And if you keep SMAD 158 shut with 4, then you just have a SMAD 4 over there, which is talking. And if you don't allow SMAD 4 to talk to 158, now where are you going to lead? So we use dorsomorphine, which can shut it. And then we have done some of the inhibition trials, and I'll keep it very brief. So this is a picture of Ocean. Ocean, if you remember there, Runex again. So in the treated and non-treated, this is how the picture came. So we moved on after the dorsomorphin picture. It did suggest that there's an involvement of BMP for sure, and other parallel pathway. We don't know what are the pathways. We came back to a picture. So is TAS, which is talking to it? We raise a question to ourselves. You remember I was talking there, a red color TAS. Whenever you raise a finger, within 3 minutes I'll close. But I think I have some time. Yeah. So then we have another independent signaling MAP kinase over there, and other signaling pathways we just don't know. We are debating it over. So then we have generated few pictures. I'll be quick. And we said these 3 things. It's independent TAS SMAD axis. And then we moved on again, generating a data. And we said, what does TAS and SMAD do? See, picture is so clear. I need not to say it. Again, we came back to our bars and we said, if you shut the SMAD4, where and TAS, what exactly is happening. So we have run some knockdown experiments, and again the cells, one can really see that what exactly is happening in TAS and SMAD4 knockdown ones, and we say it does affect the differentiation, and that's a point we raised to ourselves, and we were happy. So adipo and chondro, absolutely fine, no effect, so that's good. Then we moved on with that. So it's a rich data. If you want, I can come to conclusion. But I just like to share. It's a very nice and encouraging data. If you see Ocean Opian and RenX2 with TAS, and what you see with the gel pictures, so you would really lead to say that there is a significant reduction in the markers, and hence your thought is on a correct direction. So if you knock down TAS, what happens? Beautiful data comes, and you're very clear what happens, and you can say, yes, significant. So it markers goes significantly down. Then you come back and you ask a question. What exactly is the effect of SMAT4 and TAS knockdown to the markers when you co-transfect them? So we say that, yes, together they are playing roles. So TAS doesn't go directly to adipogenic, logs come, and TAS is continuing with the osteogenic lineages. And this is how we have done the osteogenic induction on, up to 3 weeks. And we did say that this increased osteogenic data, and we are here. It's not so complex now. We're out with that. So we just said that predominant contribution of BMP independent SMAD4 TAS signaling is actually the commitment towards the fitness of these cells. And with that, I think I just wanted to share that we then took it to the therapy also. I don't want to cover it. The published part, I started an innovative research institute where I'm translocating now. And the part which I'm showing for SMAT TAS work, which we did at NIAB, and major work was done by Ananya, and Irfan did it, Ufak Shabir. Thank you so much from National Institute of Animal Biotechnology as well as from Indian Veterinary Research Institute. Thank you very much.
Thank you, Dr. Taru, for that wonderful lecture. And you have
Thank you.
beautifully shown all the different signaling pathways and how ROS and high calcium can affect the fitness of these cells. So on behalf of the delegates and the organizing committee of Appicon 2025, I want to thank you for sharing your expertise and work with all of us. Thank you once again.
Thank you. I'll be happy if there's a question.
Yeah, I think because of
Let's-
lack of time, I think
let's
we'll
do
move
that.
on because,
Yes.
yeah, so we'll go on to the next speaker.
Thank you.
I now request the chairpersons to present the certificate of appreciation and a memento to our resource person. For the next talk on the key neurodegenerative agents in the brain, the role of endocrine disruptors, I would like our chairpersons to introduce the speaker, Dr. Sanghamitra Bandopatya.
Good morning to all of you. It's my honor to introduce our distinguished invited speaker, Dr. Sanghamitra Bandopatya, Senior Principal Scientist at Indian Institute of Toxicology Research, IITR Lucknow. Dr. Bandopatya is a leading researcher in the field of neurobiology with special interest on neurodegenerative and developmental neurotoxicology. Her extensive work has provided valuable insight into the cellular and molecular mechanism through which environmental toxins alter neuronal pathway and hormonal balance. Today, she will speak on the very relevant topic, key neurodegenerative agents in the brain, the role of endocrine disturbers. We are privileged to have her with us at Appicon 2025, and I warmly welcome her to share her expertise and findings with all of us. Please join me in welcoming Dr. Sanghamitra Bandyopadhyay.
Yes, thank you, Chairperson, for introducing me. And just before I get set into the system, because I think I'm waiting for the pointer. Yes, it's not here around. Yeah. So before doing that, point one is, yeah, there has been a shift in the program, the reason why I'm here. And secondly, exactly, fitness, it's the brain. And when toxicants, am I audible, please? Yeah. This one. You don't have this mic, collar mic? Because you get stuck up here, so that's very difficult. Do you have a collar mic, please? Yeah, please see to it, and the pointer as well. Yes, and fitness, the first aspect which I address of it is neuroscience. Yeah. Oh, it's yours. You gave it. Yeah. Thank you so very much. Yeah, it should have been here itself. Could you show how I do it? This one? Yeah. And the shifting from one to the other? No. Ah, okay. Thank you. Yeah. And fitness is brain fitness. And as my previous speaker has mentioned that in one of the slides I was seeing, she specified environmental factors. Yes, so very much that is covered because we being engulfed by the environmental problems and the neurotoxic effects associated. You don't move. Please see to it, your key is also not moving. Address the- Make it comfortable here, please. It's in day one. Yeah. You'll have a mouse? That also you don't have. Your key doesn't function. Yeah. Okay. Sorry for the interruption. Yeah. So it's the title is Key Neurodegenerative Agents in the Brain and the Role of Endocrine Disruptors. So coming to the neurodegenerative aspect, it is yes, how the neurons and the associated cell types in the brain get damaged. And today, in fact, in the truest sense, in order to fit into this concept, I have made this title as such, but it is specifically one of the endocrine disruptors and how it leads to neuronal loss, again, in a particular domain of the brain and its related effects. So me, I'm Sanghamitra Bandyopadhyay, and I'm from CSIR Indian Institute of Toxicology Research. And as you can understand that I've mentioned the word neurotoxicology several times, because that is what the toxicology is the mandate of our institute and of the 37 plus 1 CSIR institutes which we have. Ours is dedicated to the purpose of toxicology. So this is, we have to stick to it. Yes. So endocrine disrupting chemicals, again, coming to a particular one, that is bisphenol. This bisphenol, it has been found in various sources, mainly in the plastic containers, canned food liners, food packaging, and all these. But the major problem which has been pertinent in our country is finding them in the children's bottles, the baby food bottles. And the mothers have been carrying them, women have been carrying them. So it's going to be, I'm focusing on the women aspect. And of these, the prime one is the bisphenol A. So these are the different forms. And bisphenol A, the damage related to it, as well as the extent that was found in the plastic bottles and in the serum and urine levels of women and kids or children, infants, that led to a big hue and cry, leading to the ban. It's banned. The BPA is banned. So what obviously, it's no use working on a compound or a molecule which has been banned. It's not only in US, FDA first started it, and now it's in India as well. Since the reproduction, neural, metabolic disorders, cancer, etc., all, including significant brain damage was observed. So banned in baby bottles, not only as such, it's banned overall as well. So as substitutes of bisphenol A, since they add to the strength, the simplest form that could be said, to the bottles, it was difficult to come out of this bisphenol concept. So they've been replaced by bisphenol S and F, two other isoforms. But the basic structure of BPA, BPS, and BPF lie the same. So could those have a deleterious effect as well? And that is what is being addressed for the last few years when bisphenol S and F have still been allowed to continue. So coming to our linking with the endocrine concept that I've said, in the typical sense, I'm not going to talk about the endocrine hormones as such. As I said, it's specifically in the brain. And coming to the different hormones, particularly estrogen, which somehow relates to my study to the what we are going to talk about today. So interestingly, I don't know whether it's known that estrogen, it's known to be, associated with the breast, uterus, etc., the endocrine people, and it's known for all. But currently in the different regions of the brain also, there has been secretion of estrogen. Okay, it's not that it is confined only to the reproductive organs. So brain has also, though the amount is in such a minuscule that it is difficult to detect or diagnose through ELISA as well, nonetheless, it has been found. So whether the estrogen level has been affected at the sites of the brain or something related to it, that is what we started seeing. Because in general, when we checked with the serum levels, urine levels, by giving our animals, because what I'll be showing is animal-based study, rats, with the bisphenol treatments at the environmentally relevant doses. Because since, as I said, we cater to the CSIR's mandate of toxicology, we all the time have to somehow have an environmental relevance to our work or bring it to our work. And that has to be, it's better if it is in the Indian perspective. So we go along with that. And we found that, yes, I'll show that the different regions, the blood, the serum, the urine, and then again, very particularly, the brain did have bisphenol deposition when the treatment was given at the environmental relevant doses, the doses that we are exposed to, children are exposed to, and that too BPS and BPF, which are still continued in the market or in the factories. So based on that, when we found and when we started addressing, because as I said, it's women and they are carrying, the placenta has it. So when we found and checked with the estrogen levels in the blood or in the urine, we did not find any change. Okay. Nonetheless, what we found was, let's see if I have it. Okay, coming to is that why it could be that we started addressing the estrogen aspect or something related to it. It is because it's a xenoestrogen, the first aspect which led to BPA being removed from the market. So it's a xenoestrogen because of its structural similarity. They are extensively mimicking each other. So I'll show it. I'll talk about it. And not going into the structure. And these are the different effects that has been found because these bisphenols act as xenoestrogens or mimic them, and which involves the whole of our physiology, affecting the overall fitness, systemic fitness. And as I said, neurotoxicity would be spoken about. And going back to, again, as I said, the epidemiological studies, which it's not only in the worldwide, worldwide they have diagnosed it in our country as well, where it has been found in children, in the urine and the blood. I think I've said it already, why this work has been done. And coming to the brain, when this bisphenol S and F, which are currently being used as replacements of bisphenol A, it has been reported that these are the mechanisms that have had happened in the brain, where ultimately the manifestation was a memory loss. Memory loss is related to cognition, that is a cognitive damage, which is along with memory, there is a loss in rationality, etc., etc., etc. Total coordination of the memory functions and the rationality goes in a very impaired manner. The causes being inflammation, mitochondrial dysfunction, etc. And from our institute itself, one of my colleagues, he's addressing this memory loss or the cognitive impairment from bisphenol and bisphenol F. Next, so there is another important aspect, particularly when you see the ones who are acquainted with the neurobehavioral studies, One is we see the learning memory, cognitive function. The other is the motor coordination, which predominantly includes our coordination, our moving around, and along with that, the strength, the muscular strength, the grip strength. So we started addressing from my group, that whether this motor aspect, the motor coordination that I said, so the coordinating, moving around, spontaneous locomotor activity, the grip strength, they are being affected or not. So again, as I have mentioned several times, that a particular domain or different domains or regions of the brain are known to have stipulated regulated functions. So coming to the grip strength, balance posture, and the motor coordination, that is generally predominantly governed by the cerebellum. So in terms of the brain, one single particular region cannot be responsible for the total motor coordination or the total learning memory or cognitive functions. They do have overlaps, they crosstalk with each other, but nonetheless, one particular region has been defined for a specific function where that is the main one. So regarding the motor coordination, balance and posture, gripping ability, it is the cerebellum. And again, going back to the treatments that we had given, this was a strain of Vista rats. And as I said earlier, since it's estrogen, we started with the females. The female rats were taken. And I'm just showing this, my student has designed, this is the mid dose that has been given to the rats. Nonetheless, in terms of toxicology, whenever we are screening a new product or screening functions or the manifestations of a new compound, which in our case was bisphenol S and F, we are supposed to do, that is a basic principle of toxicology, we are supposed to screen at three different doses. And we did that, we did find dose-dependent effect. Nonetheless, this is the one which is the mid dose, okay? Because when we found that the 400 microgram per kg treatment to the animals were given, they were absolutely, the lethality was going up, the behavioral changes were so distinct and so very significant that it was not very conducive for the animals to be treated with. So, and our ethics issues had also been very pertinent in that. So we went into the mid-level to delve into the mechanism with BPS and BPF. And again, I repeat this several times, that this is very much within the range of the doses that we, the children, the infants are exposed to in our country. I mean, it's nothing that a dose that I've decided of the way, it has not been so. They are very much within the below the NOELs. The secreted or the excreted aspects are very much found in the blood, in the CSF, in the urine, in the serum. So that is, it's very difficult in toxicology. The basic aspect is dose decision, be it for the in vivo animals as well as for the cells. And we work on it in a very broad manner in order to come to it. So first, let me see, we first saw or explored whether when we are giving the BPS and BPF at the very known concentrations we are exposed to, is there any effect or their levels change in the brain? So obviously in the vehicle, where vehicle means it was taken in corn oil. Yes, bisphenol S and F were taken in corn oil as a vehicle. It was below detection limit. There was no bisphenol S and F visible. But both for S and F, as you see here, that the level of the BPS and BPF have been found, and distinctly why now we are exploring that why this S and F have so much of difference at the level of, despite the fact you'll see later that the extent of damage done by both of them are more or less the same. But there might be some transporter issues that led to higher deposition of BPF in the cerebellum, our region. So we we isolate different regions of the brain, and we go specifically to the site which we are targeting. And we found that it is much higher for both, significantly higher for both S and F. Nonetheless, the level is almost 10 times or more for F. So what happens to the neurons when there is so much of bisphenol S and F deposition? And as we had hypothesized, there is a significant downregulation, significant reduction in the S and F levels, which we found in the NUN level, which is a matured neuronal marker in the women's cerebellum, which undergoes a distinct decrease or significant decrease as compared to the vehicle control. This is a Nissl staining, which stains the survival or the density of the living neurons. And we found that, yes, as hypothesized, that also undergoes a reduction in the cerebellum. So now we moved on to the cells that, as such, we would like to see what is happening in vitro. We've seen in the animals. And this has a link with the, I think the dose that is shown here is a bit of a problem. So it is 1 micromolar for BPS and 10 micromolar for BPF, which relates to the amount that has reached the brain or the cerebellum when the animals were treated upon BPS and BPF. We mimicked the concentration in the in vitro work that we had been doing. This is how it was done. And we collected the cerebellar cells. And we found that it was not only the animal brain or animal serum, as we had seen earlier, that the level of the neurons had gone down. We find that there was a damage to the neurons in terms of their levels, in terms of their dendritic expression, the length, the perimeter, etc., which again indicates that, yes, it is quite a network of detrimental effects happening with the BPS and BPF exposure. Coming back to where I started with, as I said so, in terms of estrogen, we found that there was no change when BPS and BPF were given in the systemic way, that is, in the serum or in the urine. We did not find any change in the rat or the mice. Though in the human data, there have been clinical studies in children have reported it, or in women, nonetheless, we didn't find it in a rat or mice. However, could it relate to something different in our case? So when we did an ELISA in the brain, and as I said, the ELISA kits have not evolved to such a sensitive extent that a very little amount, because amount secreted in the brain is very little. And despite the fact that it goes into the blood-brain, it crosses the blood-brain barrier, nonetheless, we did not find a very significant change that could be reported in terms of the estrogen. However, we found that there was a distinct reduction in the level of estrogen receptor. So why the reason is, whether the estrogen, because yes, I'll get back to it. This is the protein data. Nonetheless, we found that there was a reduction in the mRNA levels of the ER alpha also, as well as the ER beta. So estrogen has this, predominantly 3 receptors. One is the nuclear receptor ER alpha, ER beta, as well as a G protein coupled receptor. For the G protein coupled receptor, there was no change, but it was a change in the nuclear receptors, ER alpha and the ER beta, which we found in the cerebellum of the rats, as well as in the cerebellar granule cells or other Purkinje cells, which we had isolated. So there we found that, yes, the ER alpha and the ER beta had undergone a reduction. Yes, it could be the expression of the ER alpha itself, which we are now currently working upon, rather than the estrogen directly affecting the levels of the ER alpha or the binding ability. It could be the generation of ER alpha in the brain that is being explored. So we found, then we went into the brain, and yes, again, the neurons were stained with the Newen staining. And yes, we did find that compared to the vehicle, the BPS and BPF treated ones showed a reduction of ER alpha expression, ER beta expression, or the levels in the neurons as well. This was further validated in the cells where, yes, in the primary CGN that we did isolate, upon treatment with the BPS and the BPF, there was a reduction in the ER alpha as well as ER beta in the neurons. This is the densitometry of it. Now, again, ultimately, everything happens, or you are happy or unhappy, as it might be said, when you see in front of your eyes. And that is what the reason why we go more into the learning, memory, cognition, and the motor coordination aspects in terms of the brain. We go for the retina because we do retinal work as well, where we have equipment or instruments which we use for retinal functioning measure. So these cognition and this one, the motor coordination, it's very easy to see because it's so distinct right in front of your eyes, and these instruments are there to measure them as well. So we found that upon treatment with the vehicle, compared to the vehicle, when BPS and BPF were treated, there was a distinct change in the locomotor activity of the animals, as well as in the grip strength. That's measured by the grip strength equipment. And this is exactly. So it was very significant. I mean, we have other supportive data as well, but nonetheless, this itself shows. So what the cerebellum is showing in terms of its neurons or its neuronal loss and all the aspects that we've seen, somehow it is related to, it could be related to this function, to this loss in function, that is motor coordination, which is the main or predominant function of one of them of the cerebellum. So now does the first question that comes, yes, we've observed the change in the ER alpha levels in the neurons, in vivo, in vitro, and again, we have seen that there is a neuronal loss as well as a change or damage in the motor functions. Does it relate to the ER alpha, which we have observed before? So that is what we did. We used, so the ER alpha level had gone down. So we used agonists. So as I said, ER alpha is a nuclear receptor. We used the agonist of ER alpha, which is PPT, the agonist of ER beta, which is DPN. And we found that, yes, these two agonists were capable of increasing or restoring the levels of ER alpha and beta in the brain, in the cerebellum, or rather the cerebellar neurons of this in the brain. So what we did is, how do we do it? So first, yes, we treat the BPS and the BPF, and then we co-treated. So sorry, I missed saying the BPS and BPF treatments were given through oral gavage, because that is what happens. Either the milk is being, the infants are drinking milk from the bottles, or the mother is getting exposed through these bottles. Yeah, fine. No, I'll finish less than that. So from bottles they are exposed to. So that is what our oral treatment had been done. But specifically, since we were targeting the ER alpha and the ER beta, and since it might not have been a systemic effect, so when we use the modulators of ER alpha and ER beta to link them with the functional manifestations that we are seeing, we did a stereotaxic insertion of ER alpha. This is a machine which we have. It's a stereotaxic machine where we directly, seeing the coordinates within the brain, we target the component or the molecule or the drug or the pharmacological modulator, which is PPT and DPN in this case, at the site that is cerebellum and the particular site of the cerebellum. So this one, it looks a bit crude. Nonetheless, this is what we do, or my students do the whole day. So they directly target it at that site in the cerebellum through the stereotaxic insertion of PPT and DPN, co-treated with vehicle DPS and BPS and BPF. And yes, distinctly we found that upon treatment with BPS, which we've seen earlier, there is a reduction in the newen, whereas when PPT and DPN, which restored the ER alpha level, could cause a recovery in the newen level, which is the matured neuronal marker, as we see here. The NISL, which is again the neuronal marker for survival, also shows a revival. Yes, then I think it's a repetition, probably. I think one is a cell data. This is a cell data. So one is in the CGN and the other is in the in vivo animals. And then ultimately what we found is that upon treatment with the modulators, so this is vehicle, the short form, because it was looking very cumbersome, so BPS and BPF. Along with that, when these modulators were given or specifically targeted for both S as well as BPF, PPT and DPM, they led to a recovery in the motor functions as well as the grip strength. So ultimately, yes, the conclusion is that BPS and BPF in the animal cerebellum, animal brain cerebellum, or the CGN in which we've done in vitro, there is a reduction in the ER alpha and ER beta. And then when we give the PPT or the DPN, which are pharmacological agonists of ER alpha and ER beta, they not only could cause a restoration in the neuronal ER alpha and beta in the cerebellum, but could lead to recovery in the neuronal levels and the functions of the ER alpha, and ultimately the motor functions which is predominantly regulated by the cerebellum. Oh, I think I'm going the other way around. Okay, I think I'm done with it. Yes. So it's a thank you slide. I missed, I was in a bit of a rush, that acknowledging my students. In fact, strangely, it's a women gang now. I have around 8 to 9, including me. And primarily, the work is done by my student, Uttara. She's, in fact, supported me in making the slides and finishing the whole aspect, as well as her papers are in process in relation to this. She's the highest, senior most in my group. Thank you. Yeah.
Thank you very much, ma'am, for the enlightening and thoughtful provoking presentation. Your talk clearly highlighted how endocrine disturbing chemicals like bisphenol can alter the brain biochemistry and contribute to neurodegenerative disorders, reminding us how closely environmental exposure and neuronal health are linked. On behalf of organizing committee and all delegates of Appicon 2025, I sincerely thank you for sharing your deep insight and research experience. Thank you very much, ma'am.
I now request the chairpersons to present the certificate of appreciation and a memento to the resource person. I now request Dr. A.P. Hogare, sir, Professor and Head of Department of Pharmacology, to come up on the stage and hand over the certificate of appreciation to the chairpersons.
Thank you, one and all. Thank you, chairpersons. Now we are going to start inaugural session in next 10 minutes. Thank you.
ಅರ್ಚನಾ ಇನ್ನೊಂದಿದ್ರೆ ಕೊಡ್ರಲ್ಲ ಈ ಕಡೆ ಬಾ ಇಲ್ಲಿ ಅಶ್ವಿನಿ ಆ ಸೈಡ್ ನಂದಿತಾ ಅಶ್ವಿನಿ ಈ ಸೈಡ್ ಇಬ್ರಿ ಕಡೆ ಬರೋಬರ್ನಾ ಇಲ್ಲೊಂದ್ ಮೈಕ್ ಇಸ್ಕೋದಿದೆ ಹೆಂಗಿದ್ರು ಅಪೂರ್ವ ಇಲ್ಲಿ ದೂರ ಇದೊಂದು ಇಲ್ಲೊಂದು ಹಚ್ಚ ಎಸ್ ಬರಬರ
ಇಲ್ಲ ಇದಕ್ಕ
फिर आप सेंटर में रहो एक ही साइड ओके वैसे करें
Check 1, 2, 3. Check test.
inaugural function of 71st APPI National Conference, Appicon 2025, being jointly hosted by Department of Physiology and Pharmacology, Jawaharlal Nehru Medical College, Belgavi, under the auspices of Association of Physiologists and Pharmacologists of India, APPI, with the enticing theme, Physiologists and Pharmacologists for a Fit and Healthy India. The Association of Physiologists and Pharmacologists of India is one of the most oldest and most prestigious professional bodies in the country, established in 1957. Our APPI Belgaum branch functions under its aegis and is a 7-time recipient of the Best Branch Award. Appicon 2025 has brought together brilliant minds across the country, scientists, educators, researchers, all to share, learn, and collaborate. Over the next 3 days, we shall witness keynote lectures, symposia, plenaries, poster, and oral presentations that promise to enrich our knowledge. This conference is being held at the vibrant academic environment of Keli Academy of Higher Education and Research, Kahir, a deemed to be university of international and national repute, with NAC A+ grade in its 3rd cycle and UGC 12B. Established in 1916, the Karnataka Lingayat Education Society is one of India's premier educational organizations, which is headquartered in Belgavi. With a legacy spanning over a century, the society manages more than 290 institutions across India, which encompasses schools, colleges, universities, catering to diverse fields like medicine, dentistry, pharmacy, nursing, physiotherapy, engineering, law, and management. Under its ambit, there are 9 such constituent units, which are recognized for its excellence in UG, PG, and postdoctoral courses. At its heart lies the Jawaharlal Nehru Medical College, which is a premier medical institution founded in 1963, with a proud history of spanning over 6 decades, shaping generations of doctors, educators, and researchers, and boasting of an NIRF ranking of 46. With its strong foundation in physiology and pharmacology, it is a befitting venue for Appicon 2025. Ladies and gentlemen, hosting this national event here truly reflects the institution's commitment towards innovation, collaboration, and advancement of healthcare.
It's now time to invite our distinguished dignitaries onto the dais. And to do the honors, may I request Dr. Parvati Patil, our organizing chairman, Dr. Anil Hugade sir, our co-chairman, and Dr. Harpreet Kaur, our organizing secretary, to escort them onto the dais. Today's chief guest, Professor L.S. Shashidhara sir, Director, National Center for Biological Sciences, Bengaluru. Guest of honor, Professor Benedito Machado, Department of Physiology, School of Medicine at Ribeirão University of São Paulo, Brazil. Our beloved principal, Dr. Mrs. N.S. Manshetty, Madam. Dr. Lakshmi T. Rao, General Secretary, APPI, Professor and Head of the Department of Neurophysiology, NIMANS, Bengaluru. Dr. K.P. Putturaya, former General Secretary, APPI, former Director, Rajiv Gandhi University of Health Sciences. Dr. Ashok Jarial, sir, Finance Secretary, APPI, Professor and Department of Physiology, AIMS, New Delhi. Our KMC observer, Dr. Pradeep Patil, sir, Professor and Consultant Radiologist, JNMC and Keli Prabhakar Kore Hospital and MRC Belgaum. Thank you, one and all. We stand to acknowledge the dignitaries seated amongst us. Dr. Raichur sir, Dr. H.N. Malik sir, Dr. K.K. Deepak sir, Dr. P.A. Patil sir, and Madan Godbole sir. and Mrs. Mashadu, who is there seated amongst the audience. A very warm welcome to you all.
Call upon Dr. Parvati Patil, Madam, our organizing chairman of Appicon 2025, to address the August gathering.
A very good morning, distinguished guests on and off the dais, esteemed delegates, faculty, researchers, PhD scholars, young aspiring postgraduate and undergraduate students, ladies and gentlemen. It's my honor to extend a warm and hearty welcome to each one of you to the esteemed 71st Annual National Conference of Physiologists and Pharmacologists, Appicon 2025. Hosted by the Department of Physiology and Pharmacology, JNMC Kaher, and APPI Local Branch, Belgaum, under the aegis of Association of Physiologists and Pharmacologists of India. At the outset, I would like to extend a warm welcome to our Honorable Chancellor, Dr. Prabhakar Kore sir, who in his absentia has sent his warm greetings for a successful conduct of this conference. It is an honor to welcome our esteemed chief guest, Dr. L.S. Shashidhar sir, Director, National Center for Biological Sciences, a part of the Tata Institute of Fundamental Research, Bengaluru, whose presence signifies the importance of this conference. Thank you, sir, for gracing our invitation. It's my pleasure to welcome our guest of honor, Professor Benedito Machado, Professor, Physiology, School of Medicine, Ribeiro University, Sao Paulo, Brazil. His presence emphasizes the relevance of this conference. We are grateful for making to this conference, sir. It's my privilege to welcome our principal, Madam, Dr. N.S. Manshetty, Madam. Welcome you, Madam. I'd like to extend a warm welcome to the executive members of APPI, Dr. Lakshmi T. Rao, General Secretary, APPI. Welcome you, Madam. And Professor and Head, Department of Neurophysiology, NIMANS. I also extend my welcome to Dr. Ashok Jaryal, Secretary, Head Office, and Finance Secretary, APPI, Professor Physiology at AIIMS, Delhi. Welcome you, sir. I also extend a warm welcome to our former Senior Physiologist and former Secretary, APPI, Dr. K. Putturaya, sir. Also welcome the KMC observer for the next 3 days, that is Dr. Pradeep Patil, Professor, Department of Radio Diagnosis, JNMC, Belgaum. So together we gather here for the next 3 days, united by our unwavering commitment to well-being under the theme Physiologists and Pharmacologists for a Fit and Healthy India, which underscores the pivotal role of physiologists and pharmacologists in shaping the health and commitment to advancing healthcare and well-being. We at the forefront of understanding the intricacies of human body with our collective expertise has the power to transform lives and shape the future of healthcare in India. This conference aims to share knowledge, promote research, collaboration, and innovation among experts in the field. It will surely inspire new ideas and strengthen improving health for all. Before I conclude, I would like to take a moment to acknowledge the proud legacy of APPI Belgaum branch, established in 1992, which hosted the 41st Appicon way back in 1995. Since its inception, the local branch is a pillar of excellence, fostering growth, research, innovation, and collaboration. And I'm proud to say that we have received the APPI Best Award for 5 consecutive years previously. Again, welcome to each one of you, and looking forward to a productive and enriching conference. Thank you one and all.
Thank you, Madam. I also extend a very warm welcome to our Vice Principals of JNMC, Dr. Patan Shetty sir and Dr. Rajesh Pawar sir. We take this special moment to convey our heartfelt respect and warm greetings to our esteemed dignitaries by presenting the floral bouquets. May I now request Dr. Parvati Patil to kindly come forward and present the floral bouquets. To our chief guest for today's function, Professor L.S. Shashidhara, sir. To our guest of honor, Professor Benedito Machado. Thank you, madam. I call upon Dr. Anil Hugade, sir, to present the floral bouquets to Dr. Lakshmi T. Rao, General Secretary, APPI. To Dr. K.P. Putturaya, former General Secretary, APPI. To Dr. Ashok Jarial, sir, Finance Secretary, APPI. Thank you, sir, for doing the honors. I call upon our organizing secretary, Dr. Harpreet Kaur, to present the floral bouquet to our beloved principal, Dr. Mrs. N.S. Manshetty, madam. To our KMC observer, Dr. Pradeep Patil, sir. Thank you, one and all.
To invoke the blessings of the Almighty, may I invite the team from our very own Keli University School of Music for a soulful rendition of invocation song.
ತ್ಯಾಗಭೋಗ ಸಮಯೋಗದ ದೃಷ್ಟಿ ಬೆಳವಲ ಮಲಕರ ಸುಂದರ ಸೃಷ್ಟಿ ಜ್ಞಾನದ ವಿಜ್ಞಾನದ ಕಲೆಯೈಸಿ ಸಾರೋದಯ ಧಾರಾನಗರಿ ಅರಿವೆ ಗುರು ಜ್ಯೋತಿ ವಿಶ್ವ ಭಾರತಿಗೆ ಕನ್ನಡ ಭಾರತಿ ಒಳಗಲಿ ಮಂಗನ ಜಯ ಜಯ
Thank you, one and all. To mark the auspicious beginning of this scientific gathering, I request our chief guest, sir, Professor L.S. Shashidhara, sir, our guest of honor, Professor Benedito Machado, sir, and all our dignitaries on the dais to kindly join in for the lighting of the lamp ceremony, a symbol of wisdom, knowledge, and enlightenment.
All. It's time to introduce our chief guest and guest of honor. I now request organizing president to kindly come forward and introduce our esteemed chief guest and guest of honor for this August gathering.
It's my privilege to welcome and introduce our esteemed chief guest, Professor L.S. Shashidhara, developmental biologist, geneticist, and a professor of biology currently serving as the director, National Center for Biological Science, a part of Tata Institute of Fundamental Research, Bangalore, sir. Welcome, miss. Sir has served as a professor, Indian Institute of Science Education and Research, Pune, honorary professor at Ashoka University, Sonipat, chairman of the governing board, Hyderabad Eye Research Foundation, and founder director, Pune Knowledge Cluster Foundation. Sir mainly specializes in genetics, molecular biology, and evolutionary biology. His research interests include developmental and cancer biology. After obtaining his UG/PG education at University Agriculture Sciences, Dharwad, and PhD from Cambridge, UK, he started his research career at Center for Cellular and Molecular Biology, Hyderabad. He has identified key mechanisms that specify organ development and regulate growth control during embryonic development using Drosophila fruit fly as a model system, expanding his study to examine the status of these evolutionary conserved mechanisms in epithelial cancers in human. He is a J.C. Bose National Fellow of the Department of Science and Technology and an elected fellow of the Indian National Science Academy, Indian Academy of Science, and National Academy of Science, India. For his contribution to basic and applied physiology, the Council of Scientific and Industrial Research, the apex agency of the Government of India for Scientific Research, awarded him the Shanti Swarup Bhatnagar Prize for Science and Technology, one of the highest Indian science awards in 2008. In 2018, Sir was elected as the associate member of the European Molecular Biology Organization, an international recognition for excellence in research in life science. Sir has also served as the past Vice President of Indian National Academy and Secretary General and President of the International Union of Biological Science. Sir, I would proudly like to say that he's the first Indian to be elected as the IUBS President in its 100 years of its history. During this time, he commissioned and edited an anthology on impact of science in independent India and conceived and initiated the Tropisu project, which is a transdisciplinary research-oriented pedagogy for improving climate studies and understanding, which is an international project on the climate change education. Professor L.S. Shashidhar sir, during the COVID-19 pandemic, was very active at various fronts to mitigate the impact of the pandemic all across India. Sir is serving as the co-chair member of various apex committees of the Government of India, particularly dealing with education, basic, applied, translational research, innovation, international relations, and outreach. He has facilitated training of a number of school and undergraduate teachers in adopting inquiring-based teaching methods. Sir, your gracious presence is a testament to your administration and management that have benefited more than 600 scientists from across the country. Sir has got a number of reputed publications in international journals with an H index of 26. So with this, I welcome Dr. L.S. Shashidhar sir to this forum. So today it's also my honor and privilege to introduce our guest of honor, Professor Benedito Machado, presently working as a professor in the Department of Physiology, Ribeirão Preto Medical School, University of Sao Paulo, Brazil, sir. Welcome you, sir. Sir has done his graduation in biological science, master in physiology and PhD at Ribeirão Preto Medical School, Brazil. Sir's main line of investigation is currently oriented to the understanding of the synaptic transmission in the neural pathways of the central control of autonomic and respiratory functions, particularly the coupling mechanism of these important neurovegetative functions at the brainstem level, with the focus on the modulation of the synaptic transmission by the astrocytes. His research explores the synaptic transmission of cardiovascular and respiratory reflexes at the brainstem level. His laboratory focuses mainly on the central neural control of autonomic and respiratory functions, with emphasis on neuron-astrocyte interactions. Sir's honors include, Sir is a fellow of the International Union of Physiological Science, fellow of the American Physiological Society. Sir is also a fellow of the International Union of Physiological Science and a member of the Brazilian Academy of Science. His honorable services include, he is the reviewing editor for Journal of Physiology, and presently he's on the editorial board of the Physiology Journal and the Autonomic Neuroscience Basic and Clinic Journal. He has trained a number of physiologists in this field and has published over 250 papers in international journals with a H index of 50 and contributed to several book chapters. So with this brief introduction, I welcome you.
Thank you, madam. Felicitation is all about expressing our gratitude, respect, and admiration for their dedication, vision, and excellence. Today, we take this opportunity to recognize their invaluable services. May I now request Professor L.S. Shashidhara sir to kindly come forward to receive his felicitation as a token of our deep respect and appreciation. I also request Dr. Ashok Jarial sir and Dr. Putturaya sir and other dignitaries on the dais to kindly come forward and do the honors in felicitating our chief guest, Professor L.S. Shashidhara sir. Thank you, one and all. Secondly, we also commemorate the occasion by honoring the contribution of our guest of honor, Professor Benedito Machado sir. I request you to kindly come forward to receive the felicitation. Thank you, one and all, for doing the honors.
On this occasion, I request for few inspirational thoughts and words of encouragement from our chief guest, Professor L.S. Shashidhara sir. Over to you.
Very good morning to all of you. Thank you very much, Dr. Kaur, Dr. Lakshmi Rao, Dr. Parvati Patil for inviting me for this event. I'm really honored and privileged to be here today. And also it's a bit of embarrassing to go through the felicitation, which I'm sure it was a bit of entertainment for some of you. Soon after this, I have a very long talk as a keynote speaker where I'm going to cover quite a few things about the kind of research that we do and the kind of research that needs to be done in the country to take the health of the population forward, particularly for healthy living as well as healthy aging. I just want to give a very short, brief introduction in a way, for particularly for students here, not so much for faculty. You all know the life on Earth is 3.75 billion year old now. And it is the same cellular form of life, which is continuously existing and evolving and has generated all the diversity that we see around us. The microbes, plants, animals, as well as, of course, our own existence and our own evolution and the kind of diversity that we see in the population. More so in India. India is one of the most diverse human populations living in diverse geoclimatic conditions with diverse lifestyles, cultural practices, nutritional practice, food and nutritional practices. in a country like ours with such huge diversity and also population size, 1.5 billion people, providing good health is a challenge. And health is not about treating the diseases. That's only one component of it. Preventing the diseases, preventing us to get any kind of diseases, whether infection or non-infectious diseases, is the aim of health research. And the kind of research that medical colleges do, we always think that the hospitals are more interested in treating the disease. Of course, that's the immediate goal, because you cannot wait for, for full, what called as disease-free kind of a society. And to achieve that, of course, more research has to be done. I don't know if we can ever have a disease-free kind of a society. I'll come to that in a minute, or perhaps in my talk later. Given the kind of living conditions we all live, and naturally how we are evolving, right? So the fact that we all live in a complex society, we also had to deal with variety of different kinds of health problems, right? And how do you deal with this, which is considering is such a complex thing? Forget about complexity of the society, complexity of population size, the global ones, or just forget about other organisms too. Just look at our own human body. Whatever we know about it, it came in the last a few centuries of observations. Initially, observational research, anatomy particularly, and trying to understand the symptoms of very different diseases compared to so-called normal individuals, right? Clinically normal individuals. When you do comparison, you will understand some aspect of the diseases. For example, we knew quite a bit about diabetes, although no treatment was available until the beginning of 20th century, without having understood what is the science behind the onset of diabetes and its progression in our body. But human body is made up of a variety of different organs and tissues, and each one of them is made up of several millions of cells. A human body, when it's born, after 9 months of development in the womb of the mother, starting from one cell, we are born as a 50 trillion cell body. Is astronomical number, 50 trillion cells. And there are several trillion more cells which are not differentiated but division competent for repairing the wear and tear tissues, wear and tear in the tissues, as well as for wound healing, what we call as stem cells. In addition, there are several trillion bacterial cells residing in our body, helpful bacteria, symbiotic relationship with us, what we call as microbiome. It will be gut microbiome, oral microbiome, and skin microbiome, and so forth. This collective trillions and trillions of cells make one human body. Now, if one individual, if I had to function as a one individual system, right? That system should be well connected, right? We are not simply like liver doesn't function as liver as an independent organ. Heart, brain, every one of them, they are interconnected to other organs. There are some called semi-independent organs, but they are not ever completely independent. They are connected. For example, even breathing, right? Breathing is so essential for our life. The breathing is not completely, voluntary. It is not completely independent of other organ function. For example, lung continuously help in breathing, and that is also controlled by the brain. For example, when I speak, when I walk, when I run, when I have some stress, when I want to sing at a high pitch, each time I had to change my breathing pattern. That is controlled by my brain, right? So interconnected system is what we call as physiological system, right? Physiology is also known as systems biology. Particularly in biology research, we call physiology as a systems biology. So as a physiologist, you look at the whole body as one system and not look at into a collection of different organs, right? And a cell is also in its own physiological system, right? And we call it as cell physiology. And without that, you cannot call it as a living system. So life in general is defined by physiology and not by biochemistry, not by genetics, not by molecular biology alone. DNA is the genetic material, all of you know. The chemistry of the so-called, the very essence of life, where DNA is the genetic material, how information is coded, decoded into very different functions, of the cell and overall body, is all well worked out. in biological research, what you call as molecular biology. But next part is biochemical reactions. All these proteins, when they are made, they catalyze very different biochemical reactions. It could be Krebs cycle, it could be variety of other biochemical reactions. But biochemical reaction also can happen in a test tube. For example, RT-PCR, you can do it in a test tube. PCR, you can do in a test tube. TPCR, nothing but DNA replication. RT-PCR is reverse transcription of RNA to DNA and then amplification of the DNA. You can do protein synthesis in a test tube. You can do an antigen-antibody reaction in a test tube. Each one is a biochemical reaction. Very complicated enzymatic reaction also can be done in a test tube, right? Then what is life then? If everything can be done in a test tube, then what is so different between a cell and what happens in a test tube? What happens is that the cell is several biochemical reactions are interconnected and they are regulated in a spatio-temporal manner, right? When multiple biochemical reactions are connected and regulated in a spatio-temporal manner, what emerges is physiology. So physiology is an emergent property of all the biochemical reactions. All the biochemical reactions are preceded by the structural information, that is DNA, RNA, protein, membranes, and so forth, and very different organelles like mitochondria. And what emerges is physiology. Physiology is nothing but life. So in a way, a physiologist would be actually dealing with life. A cardiologist, sorry if any cardiologist is not, any disrespect, may be dealing with heart. A lung biologist, a pulmonologist may be dealing with lungs. Someone else may be with pancreas or liver. But a physiologist deal with entire life. When you talk about healthy living, when you talk about healthy aging, when you talk about prevention of diseases, when you talk about good immune system, we need to deal with the physiology of the body that is entirely dependent on what is the genetic background in which we have born, what kind of lifestyle we have, what kind of diet we practice, what kind of, the certain kinds of prior infection, inflammation, very different types of, insult that we actually continuously receive from the environment, leading to a variety of different kinds of inflammation. That past history can have some kind of a impact later in our age. Something that happens in the childhood can have a long-term impact much later because of the epigenetic imprinting of some of those, stress factors. It could be mental stress, it could be biochemical stress, and so forth. So given all of these things, it's extremely important that we understand the physiology as a biologist, as a medical practitioner, as a public health person, and every citizen in a way, because that's the only way you can live healthily and prevent the health burden in the society. And so that the society can also live with less stress, less bodily stress, that mental stress can also lead to the more harmonious society. So with this, I'm really happy that all the physiologists, every year you gather here. I know this is a physiologist and pharmacologist conference. And pharmacologist, nowadays, it's going beyond giving medicine. It's also dealing with nutraceuticals for healthy living as well as healthy aging. So I think there's a combination of the two will keep us healthy and reduce the health burden in the society. So I really congratulate for this conference organizers for organizing this conference, this fantastic campus of Jawaharlal Nehru Medical College of KLE Society. This is my second visit. I was here when my nephew was graduated an MBBS convocation. I had come here and as a parent, and this is further a beautified place. I'm really happy to be associated with this, and would like to continue our interaction and see whether National Center for Biological Sciences, which actually looks at the biology as in its entirety, not separately as molecules or biochemistry or organ biology or development biology and so forth. So in a way, what we do at NCBS is looking at the emergent properties of the biological system, which is nothing but the physiological processes. And hopefully there will be scope for doing research with all the physiologists, on the dais as well as here in the audience. Thank you very much.
Thank you, sir. We are privileged to be graced by an eminent guest of honor today. I now request Professor Benedito Machado, sir, to deliver his address.
Good morning, my colleagues, physiologists and pharmacologists. And first of all, I wish to thank Professor Harpreet Kaur and all the organizing committee of this 71st meeting of the Association of Physiologists and Pharmacologists of India. And I must tell you that it is always a privilege for me to be in this edition of this meeting, as well as in the previous edition that I have also the privilege to attend. I need to tell you that I'm a friend of India. This is my visit number 7 to your country. Since 2004, when I came for the first visit, and then I came back for different visits in different institutes, universities, as well, different meetings. And always was a privilege, as I said. And one thing that I must let you know that in these 21 years since I came to visit India, I observe every time that I came to this nation how important is the investment in science and technology that you in India are making these recent years. I can see in the different laboratories, universities, and especially the new generation of physiologists and pharmacologists, which I am more directly connected, how important is the development of India in all areas of science and technology in the last few years. So this is, you all should be proud of the achievements of your nation, and I am happy to be in some way the privilege to follow these developments. And this is something that's really important these days, especially for countries like Brazil, my nation, and India, in which due to geographical distance, our interaction, collaborations are not so strong, especially in physiology. Of course, that these days, in terms of agriculture, pharmaceutical science, telecommunications and aircraft industry, Brazil and India are working together. But I think that we physiologists and pharmacologists, we can also take advantage of this close interaction of our nations these days in order to establish some more close collaborations in between physiologists and pharmacologists of India and Brazil. And in this direction, I can say as a member of the Brazilian Society of Physiology that we should try to organize in the future as soon as possible. I know that this is a difficult strategies to organize a joint meeting of Brazilians and Indian physiologists sometimes in the near future. I think this will bring together all the expertises of the both sides of these groups of investigators in physiology and pharmacology in Brazil and India, and you can then start some effective exchanging program of faculties, visitor professors, as well the exchange of students, PhD students, and all levels of students that I was visiting this week the BLDE University in Bijapur, and I was really impressed by the interaction with the students. In addition, since Brazil and India, we are part of the BRICS, and BRICS is another opportunity for research institutes and universities to try to establish new ways of collaboration. So with this, I am convinced that Brazil in India, in spite of the fact that we are away for thousands of miles, but we are away connected not only by the history of Brazil in India, especially Brazil, we were in the way of the Portuguese to reach India And this is one of the initial reasons that make a link in terms of the history of Brazil and India. So I am sure that this meeting of the Appicon 2025 will be a success. I will be more than happy to interact with all the faculties and students in order to make even closer this collaboration that we can make even more effective in between our nations. But again, I wish to say that what I am seeing in India these days, not only in science and technology, as I mentioned before, but in all areas of the development of the cities and the all aspects of the life of the people, that is really impressive. And you, as I said before, should be proud of the achievements of the India. And we hope as Brazilians to be part of these developments of India. So thank you very much. And I wish you a great success in this meeting, and congratulations for the organizers of this great Congress. Thank you all.
Thank you, sir.
May I now call upon General Secretary Dr. Lakshmi Rao to give us an overview of APPI and address our audience.
Good morning to everyone. It's a great pleasure to see you all in this audience. So, particularly during the 71st annual conference of APPI in beautiful city of Karnataka, which is Belgavi. Respected delegates who are on the stage and off the stage. As a General Secretary of APPI, it's my great pleasure to welcome you all to this conference, and I sincerely welcome and respect the enthusiasm and the spirit of the team led by Dr. Parvati Patil, Dr. Kaur in organizing this conference in Belagavi. Just to give you an overview about the APPI, those who are not aware of that, particularly for the youngsters, APPI was founded in 1957, led by our several senior physiologists, and more specifically some of the neurophysiologists and neurologists, like Professor B.K. Anand, Professor Baldev Singh, Professor T. Desiraju, and subsequently by our several senior physiologists. Some of the senior physiologists are here, who are the one of the few physiologists contributed significantly in the development and growth of the association, like Dr. H.N. Malik, Dr. K.P. Putturaya, and I don't see Dr. K.K. Deepak. I think there are many more senior, maybe I'm missing some of them here, not able to see them. And it is because of them that association has grown now beyond 3,000 plus members. Currently, the numbers are, I think, approximately around 3,000 plus. I think we are still growing and we are expanding also. As the General Secretary, it is my pleasure to be part of this community built over the years. It didn't happen suddenly. It took more than 7 decades to thrive in promoting focused discussions on evidence-based research findings and collaborations. APPI always aims to provide platform to our youngsters, particularly for the members and scientists, to advance research in physiology, pharmacology, and also in allied sciences. So in order to facilitate that, we organize conferences every year all over the India, and we give opportunities for keynote speakers, plenary lectures, like that several, even the oral presentation, poster presentations, we give platforms. So through this, whatever the research which we carry out in our own labs can be communicated through our community within India. Keeping the research motives in mind, the organizing team led by Dr. Parvati Patil and Dr. Kaur, with their team, has chosen the theme for the conference, which is physiologist and pharmacologist for a fit and healthy India, which is very apt for the current era. Coincidentally, today is the World Mental Health Day. And mental health comes along with the physical health. So the theme of the conference, merging with the mental health, I'm sure we will be able to build up a good, healthy India. As you have noticed, the team has invested significant time in scheduling several pre-conferences workshops. They have done it, I think, more than 10 workshops in the last 2 days. And from today onwards, so we started with the symposia, keynote lectures, plenary lectures, symposia during the conference by our eminent scientists from across the globe. The organizing team has given platform to enthusiastic young scientists, postgraduates and undergraduates by scheduling the pre-paper and poster presentation, thus valuing and respecting the time and commitment members given to the professions by participating in the conference. I'm confident that each scientific session will spark inspiration and ignite participations to renew your evidence-based research knowledge, thus efficiently enhancing research and teaching skills for the healthy India. So in the last few years, APP has taken up several initiatives and activities through collaborations. So to name few of them, we have distributing amplifiers to all our APPI members, that is called the ADA APPI equipment grants. We have allocated grants to local branches to conduct scientific activities. We also grant provisions for the event organization. We also provide partial financial support to faculty and students to attend annual conference. And we form special interest group now to trigger interact among physiologists and pharmacologists. We have very highly interactive website. It is not just displaying the activities are going. You can interact between physiologist and pharmacology. Like this, we have taken up several such initiatives. In order to grow these initiatives, we need the volunteers, and also we want all the APP members to make use of this initiative taken by the association. I sincerely thank you all in joining hands with the APPI to train our young physicians and fundamental research at the beginning itself. So as I always say, our physiologists today's physiology is tomorrow's medicine. If the physiologists are not taught properly in the beginning, I don't think it is possible to treat efficiently or maintain the healthy India. So let us all carry the momentum of this conference into our work, united in our goal to further understanding of human biology and translate this knowledge and meaningful outcomes for the society. So we have several awards. Today, we are going to announce one award that is called for the Best Medical Education and Technology Award. I invite Dr. H.N. Malik to give away this award. Dr. Farah Khalik, she's Director, Professor, and Head, University College of Medical Science, Delhi. May I invite Dr. H.N. Malik to give away this award. Yes. So one of our senior most professor, Dr. H.N. Malik, is handing over the award for the Medical Education and Technology Award in India. That's Dr. Farah Khalik, Director, Professor, and Head, University College of Medical Sciences, Delhi.
Excuse me, sir. We wish Dr. Ashok Jarial, sir, to present the floral bouquet to Dr. H.N. Malik, sir.
So with these few words, valuing your time, I conclude here. Thank you so much.
Thank you, madam. We now move on to a very special moment of this conference, the release of souvenir Epicon 2025, which is not only just a compilation of abstract scientific deliberation and messages, but also a reflection of hard work, dedication of the people involved in compiling this souvenir. May I request our chief guest and guest of honors, along with the dignitaries, to kindly release the souvenir.
It is our privilege to invite our beloved Principal Ma'am, Dr. N.S. Mansheti Ma'am, the guiding force behind Jawaharlal Nehru Medical College. Under Ma'am's visionary leadership, NMC has emerged as a center of excellence. Over to you, Ma'am.
Good afternoon, everybody. Chief guest of today's conference, Professor L.S. Shashidhara, Director of National Center for Biological Science, Bengaluru. Guest of honor, Professor Benedetto Machado. Office bearers, Dr. Lakshmi Rao, Dr. Putaria and Dr. Ashok Kumar. Conference office bearers, Dr. Parvati Patil, Dr. Harpreet Kaur. NMC observer, Dr. Pradeep Patil. All resource people who have come from all over, not only in India, but from outside also. Delegates who have come from all over the country. Students and faculty members from the other departments. I bring greetings from our Honorable Chancellor, Sir, Dr. Prabhakar Koreji, and our Vice Chancellor, Dr. Nitin Gangane, Sir, both of which had prior commitments, and that's why last minute they had to cancel coming here. However, I think without their guidance, this conference would not have been possible. The institute that you are here in today belongs to a society which is more than a century old, 110 years old almost. And the parent society has got more than 350 institutes under its ambit, not only in India, but even outside. We run a Malaysian college also here. The medical college that you're here and enjoying the campus is almost 6 decades old. And we are one of the oldest medical colleges here. So this medical college is one of the reputed private medical colleges in the country. So I am very glad that all of y'all are here to witness the 60-year-old medical college and also to enjoy our ambience and hospitality. I must congratulate actually Harpreet and Dr. Parvati Patil for having delegates from all over. I met some of them from Patna, some of them from Delhi, many of them from Delhi. So it's remarkable, I must say, to get them to a small town like Belgaum. But once you come here, you will definitely feel that you're not in a small town at all. That is what the management has given us. The ambience that it gives speaks volumes about their commitment in promoting education in this part of Karnataka. Coming to today's conference, I must say that the Department of Physiology and Pharmacology, for 3 decades, they have been conducting the state APPI and won the award for more than 5 times. So it's, but naturally that they had to conduct this national conference. So I'm glad they took up the responsibility. The theme of the conference is also very good, Fit and Healthy India. Very nice in letters, I should say. But to get a healthy India, I think it is very difficult today. The amount of problems that we have, as Dr. Shashidhar told us, are diverse in our country. It's because of the population dynamics, the diversity of our geography. So many factors are there which decide our health. And today, of course, it's our lifestyle diseases are determining everything. Most importantly, I think the brunt is on mental health. So I'm glad that Dr. Lakshmi Rao is here to share with you all the mental problems that we come across. Unfortunately, it affects the young people and even children. I'm basically a pediatrician, so we see that and the trauma that they go through, the parents and the whole family. So apart from that, of course, we have the lifestyle diseases. When we were doing physiology, we were only taught gross physiology. That time, Department of Physiology means very good teachers. And they used to teach us. I remember Dr. Narayan used to take just on the shape of the RBC 3 hours lectures for us. And tracts of the nervous system, he could draw the tracts with both hands and we used to just keep watching him. But from that stage, I think today what Dr. Shashidhar has told us is very important. We need to understand health from the cellular level. If we don't understand this, today we are, just going to degenerate. There's a vast change that has occurred in physiology. Every disease today, I should say, is explained on molecular genetics and the cellular physiology. First shock we got as a postgraduate when we had to understand simple IV fluids giving in children, how important it is to understand hemostasis. Second shock we got is when we started using ventilators, how important it is to understand the physiology of respiration well. And how does that ventilator, how it has to be modified, not only to the physiology, but also to the disease. So to understand the disease and to understand this, we really need to understand the physiology. And I think it is time that all medical colleges today need to have good labs. Genetic lab it may be, or a good physiology lab, because today what is happening is research is mainly given to the non-medical people, which is not good. Because we understand the body as a whole. So we have to motivate the younger generation to take up physiology and be there in the labs so that they will give us solutions. Because without understanding that, pharmacology is of very difficult today because it's all at the cellular level. The pharmacology is also coming down to, and the disease pathology is also coming down to. So I think we have to motivate now our younger generation students to take up this subject, know the importance of it, and understand that we need to go to the labs and do research. Because without that, I think that Make in India, what even technology, whatever we want, is not going to be possible without this motivating the younger generation. So I'm sure this conference will have a lot of interaction because listened to, I just had a few moments of the last lecture. So much of cellular or molecular level things are going on. And I know it because even in pediatrics today is most of the diseases today are at the genetic level or at the molecular level. So if we don't understand that, I think for especially the younger students today, they will never get motivated to take up this research and to move forward. Only then I think we'll have Nobel laureates also from India. So with this, I wish the conference all the success, and I'm sure the interaction will motivate all of you all. And I'm sure a lot of research is happening in our country also, which will definitely be bring pride to our nation and also to all the states in India. Thank you.
Thank you, ma'am.
May I now invite the organizing secretary of Appicon 2025, Dr. Harpreet Kaur, to propose her vote of thanks.
Good afternoon, everyone. On behalf of the organizing committee of Appicon 2025, it is my proud privilege to propose vote of thanks in front of you all. First and foremost, I express our deepest gratitude to our Honorable Vice Chancellor, Sir, Dr. Prabhakar Kore, Sir, and our Honorable Vice Chancellor, Sir, Dr. Nitin Gangane, Sir, for their continued support and commitment to organize this whole conference. Next is my heartfelt thanks to our chief guest, Sir, Dr. L. A. Shashidhara, Sir, Director, National Center for Biological Sciences. So your presence has been a lot of value to us today. And my take on this thing is, you mentioned this is my version of what you have explained to us. Our role today is not just to study the life process, but to celebrate them, to remind the society that the vitality and the balance is from the human existence. So we have taken the note of that, sir. Thank you so much for coming here, sir. So we are also deeply thankful to our guest of honor, Professor Benedito, sir. Sir, thank you for joining all of us from Brazil today, despite many challenges regarding delaying your visa process. And you managed it all with your grit and determination. And you explained the importance of collaboration, and which deeply inspired it. So I thank you so much, sir. My special thanks and gratitude to all the speakers of 2 days pre-conference workshops. We have conducted 18 workshops over 2 days. So the workshops were so well curated that the participants have given a very, very positive feedback. So a big round of applause to all the resource persons who have created the 18 workshop for last 2 days. Next, thanks to Dr. Anita Haru, Dr. Nagalakshmi, and Dr. Renu, Madam, because 3 of them has taken the responsibility, and as an organizing team, we decided that we will conduct a unique activity this year, that is PG quiz at national level. So these 3 people have helped us. Organizing committee gave all the responsibility to them, and they managed it well. So today we'll be having a national level PG quiz competition also. So big thanks to them also. The another unique feature of this conference is panel discussion, which is headed by Dr. Deepthi Kadyangri from Community Medicine and Dr. Aran Raichur sir from USM KLE, Dr. Utpal sir from West Bengal, with a unique theme that we have 2 YouTubers with us who are teaching the physiology through YouTube versus how it is going to be the Blackboard teaching. So we have Dr. Preeti Gupta and we have, sir, I can't see him, but yes, thank to all the people who have organized this whole panel discussion. Thank you for all speakers for the today's symposium, all invited speakers and plenary speakers for graciously accepting our invitation. And I don't know whether my stars were good this year while organizing this conference. Whomever I have contacted, I think everyone graciously accepted the invitation in one call only. even the delegates, whoever we called for anything, anybody I have called in whole APPI family, I can say, everyone had cooperative, everyone was very, very cooperative. So I have to thank each and everyone along with the organizing committee. So as in part of, everybody was talking about the importance of exercise today. So I would like to, end my word of thanks with one small activity. If all of you can raise your right hand up, take it back if you can touch your own back and pat yourself to be here and be the part of this conference. Thank you so much. Thank you so much, one and all. And I also thank Dr. Pradeep Patil, sir, KMC Observer, to be here. Thank you, one and all.
Thank you. As we reach the end of this formal inauguration, we thank our dignitaries as well as delegates for gracing this occasion. So I request the dignitaries on and off the dais along with delegates to kindly rise for the national anthem.
तव शुभ में जागे तव शुभ आशीष मांगे गाहे तब जय गाथा जन गण मन जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you, one and all. Give us a couple of minutes. We shall be starting off with the first talk of the day, keynote speaker.
Caller mic ಕೊಡಿ. ಜರಾ ಕರಿ. ತೆಗೆದು. ಇದೆಯಲ್ಲ ಅದನ್ನು ಆಯ್ತು ಹಿಂಗೆ ಇರ್ಲಿ ಆಮೇಲೆ ಮಾಡಿ ಅವ್ರು ಬೇಕಾದ್ರೆ ಈಗ ಹಿಂಗೆ ಇರ್ಲಿ ಆಯ್ತು ಇನ್ನು 2 ನಿಮಿಷ ಇದೆಯಲ್ಲ
I request everyone to please be seated. We are going to start for our next session in just few minutes. May I have your attention, please? I request all the delegates to please be seated.
We request the delegates to be seated. We shall be starting shortly. We'll start with the very first talk, our keynote address.
Now, moving ahead with the series of session, it is my honor to invite our esteemed chairperson for this session, Dr. R.N. Raichur, Professor and Head of Department Physiology, USM KLE International Medical Program, and Dr. P.A. Patil, Professor and Head of Department Pharmacology, USM KLE International Medical Program, to kindly come on stage.
ತೆಗಿತಾ
Hello. Hello. Both, huh?
I request all the delegates to kindly be seated. I will.
Aye. Now I request our chairperson, Dr. R.N. Raichu, sir, to please introduce our Professor L.S. Shashidhar, sir, for the topic Deep Tech Innovations for Affordable Healthcare for All.
Hello. Good afternoon, everybody. It gives me immense pleasure to introduce our distinguished keynote speaker for today, Professor L.S. Shashidhara, who is working as Director, National Center for Biological Sciences, Bengaluru, and served as Professor, Indian Institute of Science Education and Research, Pune, Honorary Professor, Ashoka University, Sonipat, Chairman, Governing Board, Hyderabad Eye Research Foundation, and Founder Director, Pune Knowledge Cluster Foundation. Coming to common areas of his specialization, Professor Shashidhara specializes in genetics, molecular biology, and evolutionary biology. After his PG education at University of Agriculture Science, Dharwad, and PhD from Cambridge, UK, he started independent research career at CCMB, Hyderabad. His group has identified key mechanisms that specify organ development and regulate growth control during embryonic development using Drosophila as a model system. They've also expanded their study to examine the status of this evolutionarily conserved mechanism in epithelial cancers in human. For his contribution, he is recognized with CSIR Technology and SS Bhatnagar Prizes and JC Bose National Research Fellowship. He is a fellow of all three science academies of India. In 2018, he was elected as associate member of European Molecular Biology Organization. He is one of the founding academic member of Indian University of Science Education and Research, Pune, and Ashoka University. He has served in the past as Vice President of Indian National Science Academy and as Secretary General and President of International.
No, you already introduced as the chief guest exactly the same way. It's okay, Dan. Thank you. Shall I start?
Okay. Okay, sir. So we are greatly honored to have among us such an eminent, distinguished, renowned researcher and globally respected authority. And we are indeed privileged to have such a towering personality to share insights with us today. Let us all welcome Professor Shashidhara with a big round of applause. Thank you.
Thank you. Thank you very much for the kind introduction. It's a pleasure to be here. So although I'm not directly working in the area of physiology or pharmacology, as a biologist and as a science administrator, I have some understanding of what is to be done in the country and what are the very different policies that are coming out. For example, Fit India, which the morning session was all about. I guess there is so much to discuss in a conference such as this one. So I'll sort of briefly mention a few of the initiatives, some of them initiated by us in our own institution and in partnership with other institutions. Large number of them are actually clinical research involving clinicians. Some of them are also public health research involving hospitals as well as public health officials in the country and very different policy bodies. So the title is more about what kind of innovation we need. Having understood the knowledge, how to apply the knowledge to solve real life problem is what innovation is all about, right? So innovation is not simply application or translation. The reason innovation is somewhat different than the other two is innovation, when looking for a solution, you more or less, you know what the solution is. You already know the knowledge behind the solution, science-driven solution, but that solution has to be made acceptable, affordable, accessible to the population. And so innovation that is done in US or innovation that's done in Europe could be a different kind of a parameters we set in. When innovation has to happen in India, our parameters are somewhat different in terms of the acceptability, affordability, and accessibility. So, but it has to be depend on the science, the most advanced science. Most advanced science is also coupled with technology, deep technology, and that's why I mentioned deep tech innovation for public health, affordable healthcare for all. When you say affordable healthcare for all, obviously it has to be affordable and accessible. Accessibility is also equally important. For example, all of you know, during the COVID time, the Covaxin and Covishield, accessibility is much easier because even could go to remote places without deep refrigeration or anything. It was easy to, administer vaccine. For example, if you had dependent on, let's say, Moderna mRNA vaccine, which requires, deep freezer facility, even, to take it to the remote places for administration, which is very difficult in Indian context. So when one has to do innovation, we have to look at all of these, issues surrounding this. I just mentioned in the morning, in the inauguration one, about the chemistry of life. All of you know chemistry of life is identical across all organisms, from the earliest form of bacteria-like organisms to today's more modern, recently evolved species. It could be a plant or animal or insect or human and other mammals, right? And this chemistry of life has enabled us to understand very different aspects of cell biology, organ biology, organism biology, and population biology. And at the same time, also help us to identify appropriate pathways or specific proteins as targets for any therapy, right? And that's why there has been an explosion in the biomedical research and developing new therapeutic avenues, both diagnostic and therapeutic for diseases post molecular biology era, that is 1960s onwards. Now, I just mentioned in the morning about what is physiology. The physiology is an emergent property of very different biochemical reaction happening in an organized way inside a cell. Cell itself has certain boundary conditions. It's an organized system. In an organized system, varied biochemical reaction happening in different parts of the cell. And when they are interconnected, and what emerges is called as life. And that life is nothing but the interconnectedness of the biochemical reactions in time and space. And in other words, it can also be called as systems biology or physiology, right? What is most important is every time DNA replicates, I think I have a slide on that one, it's not identical. One of the most important aspect of life is information when it is copied from generation after generation, when DNA is copied and transferred to another generation, it has to have a high degree of fidelity. Otherwise, life cannot be sustained on Earth so easily. For example, all 7 million people, we are born to our parents, and we are identical in terms of our anatomy, our, physiology, and so forth. There could be some diversity in terms of color of the skin or height or weight or some of the physiological features, also maybe some small variations are there. But as far as members of the same species, we're all born as human beings, right? But that level of fidelity, 100% fidelity, is also deleterious in a way, because the environment is continuously changing because of the physical parameters changing, as well as the biological system also change the environment. So we had to adapt to changing environment. Adaptation to changing environment happens if there is sufficient genetic diversity in the population. If you have lived for 3.75 billion years as life on Earth, or 200,000 years as modern human species, which were most recent one, like Homo sapiens, it's because of these small variation that happens during copying mechanism, which is tolerated, and that tolerance in the errors in the copying mechanism generate variations for adaptation. But at the population level, these variations are very important for continuum of the species, generation after generation for thousands and thousands of years. But for individual, it's a bane, because in the individual, I may have certain variations which may cause some diseases. So may cause some insufficiency or less efficient immune response to infection, right? We are, for example, our glucose metabolism may be less better controlled. So that's the bane of individuals, and that's what we need to address in the public health system. But we cannot avoid. That's one of the things I was mentioning in the morning. Certain kinds of health-related issues, it's unavoidable, because without these variations, we would not even existed today as human species. We would not have continued as a human species for so many years, thousands of years. But at the same time, some individuals do get affected. For example, cancer is one of those. It's a stochastic event. Some mutations may occur in somatic cells, and certain kinds of physiological variations in the body system may allow cancer to emerge as a disease. In some people, simply it comes as a benign tumor and it'll die down within no time. So this is what we need to address continuously as a health problem in the society. But there are some are preventable, some are not so preventable, right? That's the whole thing. But you can actually help in reducing the burden with better understanding of the system. Just give you an example how modern science has helped to develop better diagnostic and therapeutics, which all of you know, but this is one of the most telltale example of what science can do to society. Until 1920s, there was no really treatment for diabetes in any of the traditional system. People knew what is diabetes is. People knew, particularly in the late 19th century, we already knew it's increase in the sugar levels in the body, glucose levels in the body fluid. But we didn't know what the therapy is. The person could be a king or a queen, or it could be a servant or a soldier. They would all have the same fate if they are diabetic. But with better understanding of evolution, people understood that not only anatomically, mammals have certain similarities, physiologically also they have a lot of similarities, right? Now, of course, we know even better to an extent that we can take wall of heart of a pig or the whole heart of a pig and use it for transplantation to human. It can work fantastically. But anyway, by 1920s, we had sufficient understanding of the similarity in the physiology amongst all mammals. And also the later when proteins were discovered, and one of the proteins that was discovered and studied in the early days was insulin, and insulin is required for glucose metabolism, right? Controlling the glucose metabolism. And that insulin is required for glucose metabolism across all mammalians. And that also was understood by 1920s. So first thing, at that time, a company called Lilly, now it's called Eli Lilly because of the merging of the two companies. What they did was to extract insulin protein from slaughterhouses of cattle and horses and other animals, and lyophilized and put it in a tube and sold it as anti-diabetic therapy, particularly for type 1 diabetes. Now, as you could make out, the only small amount of insulin was available from such large animals for extracting and then giving it on. So suddenly, while treatment was available, it was very expensive. So when diabetes was considered as no cure disease, and now diabetes became a rich person's disease, right? So because cure is available, but it is not affordable. One of the things that happened in particularly in middle class families in India until 70s and 80s, many of you may know in your families, you ask your grandfathers or granduncles, that once the cure is available, the tendency is to somehow get the treatment to your, the family member who is affected, right? You cannot simply keep quiet, no, it's not affordable, I can't go to the hospital and get anti-diabetic therapy. Somehow you want to get it. You sell your house, sell your land and everything, and you want to get cure for your family members. So in a way, we went through a difficult era all over the world. It's not India specific for over 50, 60 years. Then even better understanding of chemistry of life. We know now that at the chemical level, there's no difference between a bacteria or a human or a yeast. You can take a piece of human DNA, let's say coding for insulin, put it in bacteria or yeast. What yeast will do is transcribe and translate exactly the same nucleotide sequence. What comes out is insulin protein. The yeast or bacteria may not have an insulin protein of its own. Evolutionarily, they may not have evolved at that time, but what they produce will be insulin protein. That insulin protein you can purify and give it as, as a diabetic therapy. Now, insulin, recombinant DNA-based insulin, which is lab-made or factory-made insulin, is available at such low cost. In most government hospitals in India, it is actually given free of cost. So now it is affordable, accessible, and now there is equity in treatment to diabetes, right? So that's what the science can do in the society to solve real life problems, right? But it took about 150 years. It's not that, it happened overnight. But now things are happening much faster because the way information is generated, how knowledge is created, how knowledge is disseminated, how knowledge is, used for solving the problem is all happening much faster rate because a very large number of people are involved in research as well as in translational aspect of it. And at the same time, there are tools available for almost real-time dissemination of knowledge across the world. Now, we are dealing with human body. As all of you know, in the morning also mentioned, we all are born as one cell. It is basically a fertilized cell, fusion of the egg and the sperm. And from one cell, at the end of 9 months, what comes out is at least 50 trillion cell body. In addition, there are so many stem cells. I'm talking about 50 trillion cells which are constituting the functional aspect of the body, particularly the structural part, right? And this one, from a single cell when it comes, what are the different changes it goes through, right? From one cell to multiple cells, then their positional information. For example, eyes are exactly in this place, ears are exactly in this place. It happens reproducibly across all 7 billion people in a very programmed way, because genetically programmed. And also the size and shape of organs are, determined proportionate to the body size, again, in a reproducible way. And all of these organs and tissues are physiologically connected, finally, to come out as a living individual humans, right? And these are the changes that go through during development, because it's a single cell, whether a neuron or a muscle, which look like so different, with completely different kinds of cells, but they're actually origin from the same common human cell. And so what happens is some of the internal cellular structural changes, and then complex regulatory circuit for cell division, because one cell, when it divides, it has its own control mechanism, what called cell cycle phases. But when multiple cells have to divide in a synchronous way, the regulatory circuit become more complex. This is where, again, physiology emerges. When multiple cells are interconnected and then divide in synchronous way, that's a physiological process. As if a single cell divides, we simply call it a cell division, right? Then at the reductionist level, because it's single cell, gives 50 trillion cells through mitotic cell division, all the cells will have exactly the same genome. And that means what distinguishes between different cell types along the body axis are because of the spatiotemporal regulation of gene expression, right? And the eukaryotic gene expression is a very complex process in itself. It's extremely well regulated. For example, if eye is formed here, because eye forming genes are expressed only here and not expressed anywhere else. It's both positive control as well as negative control, because even by mistake, you can't have an eye somewhere else for a better functional part of the body, right? So all this information is already encoded in the DNA, and it is sequentially very different genes are expressed in different parts of the body along the time and space during development. So I'll just skip some of these things, cell cycle phases, and these are how cell decides when to divide, and also what are the checks and balances of cell division and so forth. But when multiple cells are involved, the regulation becomes more complex, and large number of signaling pathways are involved, and these signaling pathways keep cells are extremely well controlled. Why am I giving so much of details? The reason is any perturbance in this one, more the complex regulatory system, and you have multiple different ways of controlling it. Something can go wrong. When something can go wrong, there can be some turbulence in the system. The turbulence in the system, what we call as human diseases, right? And that's precisely what happens, whether it is glucose metabolism related, whether it is cell division related, whether it is immunological system related, inflammation, or kind of thing. Inflammation is a necessary step in our body to protect from very different foreign, it could be a cancer cell, the immune response to against cancer, it could be infection, it could be pollution. There is always some inflammation. But if inflammation happens beyond certain levels, if it is not well controlled, inflammation also has so many checks and balances to keep it under control. But if over inflammation happens, that is a problem. You call it as some kind of a disease condition. So I already mentioned there are some cells are called stem cells. These are set aside for, let's say, wound healing or repair of worn out tissues and so forth. Now, presence of stem cells is a boon because we can live longer because our cells continuously functioning, whether it could be organs like a heart, which works 24/7, lung works for 24/7. But let's say if I live for 80 years, the amount of stress it has to go through, mechanical stress, simply like, other than the other stresses, reactive oxygen stress and other things, you forget about it. Simply look at the mechanical stress itself. So that definitely there will be wear and tear. Our stomach, for example, amount of work it has to do. First of all, we eat so many toxic stuff, and then we insult our stomach. At the same time, there is also mechanical way of digesting the food. So it also undergoes a lot of stress. So wear and tear is a common thing if you live longer. So thankfully, because of stem cells, we do have a way to live longer and healthily. But at the same time, having stem cells also gives another problem, which is cancer. These stem cells, if they're not controlled during cell division, it becomes uncontrolled, it may lead to cancer. But cancer won't happen just simply like that, simply because cells are dividing faster. There are various checks and balances to keep the cell division under control. Irrespective of all of these things, unfortunately, few individuals may develop cancer, right? As I'm speaking, right, at every minute, there will be somewhere in my body, a few stem cells may be going rogue and developing as some kind of a tumor. But the tumor will not lead to cancer because there are sufficient steps in my physiological processes to keep and removing the tumor cells. But sometime, if I have a problem in variety of physiological process, I may actually end up developing cancer. Because we live longer, there are multiple number of cell division happens, there are trillions of stem cells, and also there are 7 billion people on Earth. The number of patients with cancer is increasing simply as a probability of numbers, and not because of any other reasons. And of course, so address all of these things. This is one of the major solutions we are thinking is to comprehensively address cancer and not simply looking at one gene and see whether we can use this as a target, develop a small molecule therapy and so forth. That kind of a reductionist approach is not working very well. Most people also are chemo-resistant. Like antimicrobial resistance, bacteria develop antimicrobial resistance, our cancer cells also develop chemo-resistance. Because let's say there are million cells you are targeting with some therapy, a few thousand cells may actually escape, the therapy, and then next time when you give second dose, they would already become very resistant, right? Those are the kind of problem. So to address this comprehensively at all levels, using genomics, multi-omics, physiological, and using diet and lifestyle factors and everything, we have developed a consortium called Indian Cancer Genome Atlas. There are so many institutions that are involved, and we're also, getting into this big project. And perhaps some of you can join this consortium and contribute to cancer research in the country. And our primary focus in the short term is to address India-specific cancer problem. One of them is actually breast cancer. And breast cancer, oral cancer, and ovarian cancer is something which you are focusing on. Like, for example, the TNBC, triple negative breast cancer, is more of an India-specific problem for a variety of reasons, and that we need to address first. Second one is, we also have another program. Shankar is here, is within collaboration with NIMHANS. It's on psychiatric disorders, right? There are a variety of reasons individuals, because of genetic reasons or for other reasons, may develop certain kinds of psychiatric disorders like schizophrenia, bipolar, and other things. And we need to understand this again at the system level, not as some genes mutated and some disease happens. That is a very reductionist approach, particularly for diagnosis and therapy. It's not going to help, right? So to do this, we are actually now developing organoid models, the diseases. You generate brain organoid from take the patient cells and develop induced pluripotent cells, and then you generate organoids. Those organoids, is like a small brain, and that from a patient versus the normal individuals from the same family, you can compare because they have similar genetic background, similar kind of socioeconomic background, and then why one person has developed, one person has not developed, and that will help us to how to address the problem where there is a disease, right? So that work is going on in our Center for Brain and Mind in our institution. And as you all know, we recently announced India sequenced 10,000 human genomes representing large communities of India from across the country. But very soon, we are going to launch another phase. We're going to sequence 1 million individuals representing more or less every ethnic group in India. And that genome information is extremely useful to understand human genetic diversity and what are the risk factors, genetic risk factors associated with diseases. And then we can actually look at the metadata available based on the geoclimatic, nutrition, lifestyle factors which are influencing the diseases. We can actually develop a fantastic way and a metrics to understand what are the risk factors for diseases and how to reduce those risk factors so that we can reduce the health burden in the society. So Genome India project is also very, very important. It's a very ambitious program of the country. Now, I'll skip this. I already spoke about inflammation. The inflammation is largely some genetic factors. Very often it's because of the prior exposure to infection, pollution, and variety of other factors, right? And originally what inflammation was considered as something very essential factor which is required for our body, which is true, but large number of individuals are going through one or other problems because of over activated inflammation, right? And this has to be addressed in a systematic way. It's actually a physiological problem, and this needs to be looked at the physiological level and not at the molecular level. So we had to look at very different pathways involved in how the immune, particularly innate immune system, responds to very different stimuli, and then why so many individuals are affected by the overactivated inflammation system. Other one is we have not studied much, which I request, if possible, many of the people who are coming from different hospitals and public health and community health experts here to contribute to understand the immunological diversity at the population level. So, as you can see, our immune system is one of the most important to keep our, body in a healthy way, right? We cannot invest so much of our energy to fight any infection, pollution, and some of the internal factors which will cause inflammation all the time. So we need to keep immune system also healthy and reduce the amount of energy, the resources it uses in the body for fighting the system rather than using for other activity, physical activity or cognitive ability activities and so forth. Particularly among the children, if immune system is not functioning properly, if there is a over inflammation and in the lung or in other places, invariably they will have lower physical and cognitive abilities, which will affect their entire, future life. So to understand this, we need to do at the population level. COVID gave us an opportunity to do this. Some of us in Pune, in Bangalore, and in Hyderabad initiated a long-term longitudinal study to understand how immune system is functioning in response to vaccination and prior infection and for SARS-CoV-2. This kind of information for different kinds of infection and also for autoimmune diseases will help to generate sufficient amount of data for public health purposes. And this has to be done in every part of the country. No 1 or 2 cities will be enough. Even if people are coming from different parts of the world, for example, Bangalore is now, every state is well represented in Bangalore. I should be able to genetically identify every ethnic group in Bangalore itself and study them, but that's still not enough. We have to look at in the local region also, because there are many of these factors also depend on their local environmental food, what kind of food they eat, how the food is grown, whether it is grown using chemical fertilizers or organic food production and so forth. So there are a variety of factors which influence the health factors. It's a challenge. These are the things which are now undergoing. So with ICMR, hopefully we'll initiate a large-scale immunological research program, and many people can participate in a cohort, to run a cohort. But it has to be interdisciplinary, interconnected, and multi-institutional program, because we need to deal with large number of people. As you all know, because of the climate change, variety of things is happening, and it's affecting the health, particularly both physical and mental health, and also incidence of zoonotic diseases like SARS, COVID is also increasing. To address this, we had to go through what's called One Health approach, and not simply looking at only humans and treating them humans. We need to also try to address our environmental problem. We show that the environment in village is also healthy. not just we are healthy. We will be healthy only if our surrounding environment is healthy. So we need to look at animals, plants, and water, everything, and then see how we can reduce the health risk and prevent the epidemic outbreaks. There are multiple methods to continuously monitor the One Health, monitor the environment and everything. For example, we have been monitoring in Bangalore and Hyderabad. Even today, SARS-CoV-2, this is only up to December 24, but as we are speaking every day, we are getting samples from the municipal corporation and see whether there is any SARS-CoV-2 virus in the sewage water. Why is the sewage water SARS-CoV-2 will be there? Because some individuals are infected and they shed the virus. Because of vaccination, there may not be any clinical reports. For example, what is shown in the red is in the clinical. And for the last 2 years, there are hardly any major clinical outbreak of SARS-CoV-2. But it is there in the circulation. It's there in the human body. It is continuously studied. But sometimes it can be much, much larger amount of SARS-CoV-2 in the population. Thankfully, because of vaccination, it is protected. The process of vaccination is actually a physiological process. And the vaccines work, vaccines protect, vaccines prevent diseases. It may not prevent the infection. It is not meant to prevent the infection. It is meant to prevent diseases, right? And it is happening. So if there are any vaccine skeptics out in the population and public, I think please do talk to them about how well the vaccines have worked. And this is the best, evidence to say that vaccines have worked, because the amount of SARS-CoV-2 in the population is very, very high, much higher than what it was before vaccination. We are also doing wastewater surveillance for other respiratory viruses in Bangalore and Pune, which will also keep an eye on in case there is any new variant is emerging, which may create some kind of havoc. And as you all know, antimicrobial resistance is going to be another major problem. It's already a major problem in India and globally, and particularly in India. Millions of people are dying because of, sepsis and variety of different kind of, antibiotic resistant infections. And we need to understand the science behind this and also do continuously surveillance. For example, if any new antibiotic resistant bacteria is emerging, what kind of resistance mechanism they have evolved, and how to address this problem substantially. There is one is prevention. Prevention is more scientific use of antibiotics, less use of antibiotics, unavoidable. You should use it only if it is completely unavoidable, rather than simply use the antibiotic as the first course of action. And it should be the last course of action rather than the first course of action. And then, of course, continuous monitoring in the wastewater and in the clinical level. And that's exactly what we are doing, and we're generating huge amount of data, which is useful for the public. Hundreds of people in this consortium are working on AMR in our collaborative group. I'll skip this. The future of medicine, the last 2 slides. As you all see, it has to be more personalized, right? So that, unnecessarily, giving drug at a higher dose and less efficient drug, just the whole population is going to increase the health burden of the population rather than finally getting rid of the diseases. Personalized medicine is the key, and which was initially it was less possible is because we didn't understand enough about the genetic diversity or other physiological diversity of human body. Now with better understanding, personalized medicine is possible. Although for 25 years, people have been talking about personalized medicine since the human genome was sequenced, but it is becoming possible only in the recent days. And there are so many challenges too, because of the increased lifespan. Some new diseases are emerging at a larger scale. Dementia, Alzheimer's, Parkinson is also becoming much larger in the Indian country because of the aging population. We are taking the benefit of the demographic dividend today because we have largest population of young people. Unfortunately, in another 25 years, when India reaches 2047, so-called 100 years of independence, at when we want to be in Vikshit Bharat, but the challenge to reach Vikshit Bharat is all about what is the per capita GDP and what kind of a thing, what is the health index of the people. And unfortunately, we may reach a stage with largest number of old people in the world. We may actually have an increased health burden unless we take some preventive measures now itself. Okay, one of the things for that improved health on the, as I tell, healthy aging is so important, so that by 2047, when we think we are in a stage where we are in a sustainable living, healthy living, and the population level with less health burden, we need to today itself start monitoring the physiological processes continuously. Gadgets are available. You're all wearing wearables, but wearables have to be further improvised, made it more affordable. Currently, some of the wearables are, very expensive. I don't think poor people can afford that. So we need to develop better wearables of measuring variety of different kinds of physiological parameters, non-invasive sensors at low cost, particularly with quantum sensors. For example, they're going to be much more sensitive, much more informative, but you have to make it affordable. And that's the challenge for the next 10 years, where physiologists and the technocrats have to work together to develop these physiological sensors so that you continuously monitor individuals as they age so that you know when to intervene in case there is a problem before it becomes a medical problem. That's only you have to treat them, right? And that's how the treatment can be using some kind of a non-toxic therapies, right? It can be simply modification of lifestyle or diet so that physiologists can actually advise them well in advance before it becomes a medical problem and you are continuously monitoring. That's the future of the medicine. And towards this, India has launched a major biotech policy where understanding of biology and developing technology, what called biotechnological interventions, to understand the health of individuals is so important, and that helps in preventing the major disease outbreaks, whether it is communicable or non-communicable. And one major initiative in that direction, which started by TIFR, our mother institution, NCB is also a partner to this, and it is what is called Advanced Research Unit on Metabolism Development in Aging. Every individual, irrespective of medically they are in certain health condition or not, should be monitored with certain metabolism and metabolic pathways, so that incremental changes or turbulence that happens is immediately noticed, and so that one corrective measures can be taken with the help of better nutrition nutraceuticals. That's the aim of this particular center, and we are already working with using different cohort, one in Karnataka, one in Maharashtra, and few other places in other parts of India. So this has to have at least 100 cohort representing more or less all regions of India. I'll stop here. Thank you very much.
Thank you, sir. We now request our chairpersons to kindly hand over the certificate of appreciation to our speaker.
Thank you, sir, very much for your informative lecture. I think you started with right from the bacterial cell and how it has evolved. You also mentioned about the Genome India project and other things. And what I feel is physiology being the foundation of all the health sciences, it needs to be still probed the basal level. Pharmacology, you told, that personalized medicines are now that concept. It is not new one. It was introduced earlier and it is being worked out. And for that, we need personalized physiology. Without physiology, personalized physiology, you cannot evolve personalized medicine. Very difficult. And this Genome India project, if it becomes a reality in the future, I think it may help to prevent a lot of drug-induced adversities and others. Then you also told some of these old age diseases like dementia, Alzheimer's, and even that Parkinson's disease, where research needs to be still strengthened. There is no remedy for that. Permanent remedy, I should say. Temporarily they may live. I think these things, I think because it is a keynote address, I don't think questions will be entertained. Anyway, he will be there with us and you can interact leisurely if you have got it. Thank you very much, sir.
Thank you, sir. I now request Dr. Parvati Patil, ma'am, Professor, Head of Department, Department of Physiology, to kindly hand over the certificates to our chairpersons. Thank you, ma'am. Moving next to the plenary session, it is my privilege to invite our distinguished chairperson, Dr. Madhav Prabhu, Professor, Department of Medicine, and Dr. Padmashri Kudichi, Professor, Department of Physiology, to kindly come on stage and preside over the session on the topic Novel Strategies to Treat Endogenous Depression Induced Abnormal Structural and Functional Plasticity Leading to Cognitive Deficit by Dr. B.S. Shankar Narayan Rao.
Good afternoon, everyone. It gives me immense pleasure to introduce the next speaker, a renowned personality in the field of physiology, Dr. B.S. Shankar Narayana Rao ji, Professor, Department of Neurophysiology, Faculty in Charge, Electrophysiology Laboratory, Officer in Charge, Central Animal Research Facility, CARF, National Institute of Mental Health Sciences and Neurosciences, NIMHANS, that is Institute of National Importance. He's going to speak about novel strategies to treat endogenous depression-induced abnormal structural and functional plasticity leading to cognitive deficits. A very important topic, given the fact that we are having a lot of geriatric patients now, and many of the patients coming to us nowadays are coming with cognitive deficits. As a physician, this would be a very interesting topic to me.
So thank you very much for the kind introduction, and also I want to thank the organizers for inviting us to attend the meeting and also to share. Today is a World Mental Health Day. So October 10th, we celebrate the World Mental Health Day. So keeping that in mind, so I'll be talking about the mental health issues. So mainly the depression. Since from the morning, we have been listening that, okay, the physiology in totality, we need to understand the body as a whole. So in that perspective, we need to have a complex understanding of the disease mechanisms and try to see that is it possible for us to evolve the novel therapies. So that is very, very challenging when it comes to the mental health issues. So the depression, the major depression is a serious recurrent disorder, which is associated with diminished functioning of the quality of life. The depression has a lifetime prevalence of about 9.1% in India. Now, we are conducting the nationwide, the mental health survey. The numbers is going to emerge in a couple of months' time. So we will get to know that what is the prevalence in India. And the worldwide prevalence is about more than 15% or so. What is most important is that it affects the individuals in the mean age of 24 to 40. That means when they are in a highly productive age, so if they get into the problem of depression, and it is going to have a serious impact onto the productivity. So depression severely affects the structural and functional integrity of the three important brain structures, so the prefrontal cortex, hippocampus, and amygdala, and resulting in mood changes, behavioral alterations, and impaired attention, and also cognitive functions. So what is very challenging is that despite several decades of intense research on understanding the etiology associated with depression, we are not yet close to understand the precise pathophysiology of depressive disorders. So it comes with a multiple factors. So those factors need to be understood in a greater detail if you want to understand the disorder as such. So major depression is a mood disorder which is characterized by sustained disturbance in emotional, cognitive, and behavioral and somatic regulation. So what is very interesting is that now these days in the newspaper, you get to see that a large number of students, they are under different kinds of anxiety and depressive conditions. You get to see them. So a lot of the individuals at the early young age, so there are various factors which will impinge on them and they will get into the state of depression. And many of them, we get to see that they commit suicides as well. So something which begins with a lack of interest or lack of a pleasurable experience, and it translates into a lack of concentration, fatigue, and you have the sleep alteration, guilt and worthlessness, and that drives the individuals into the suicidality. So it doesn't mean that it's going to start, in a day or so. So there are several things which will be bothering them for over a period of time, which will translate into a major depressive disorder. So among several dysfunctions, the cognitive dysfunctions are the most important one, because your attention is altered, your executive functions and visuospatial functions has been altered. More importantly, day-to-day activity like working memory and also the declarative memories have been affected in the major depressive disorders. So for us to understand the depression using the human subjects, it is very, very challenging. That means once you have the depression or they are in the verge of depression, you are going to diagnose and we are going to image the brain, we are going to understand the brain circuits, how it has been affected or something like that. But we need to understand the disease process as such. So that means we have to heavily depend on the animal models, and we need to establish the animal models which mimics the human condition of the depression. So here we study the human endogenous depression. So where developmentally, if a serotonergic system has been affected, and when these individuals, they become adult, they are more susceptible for various forms of the depression. That's what we study. So what we do is that, in the crucial phase of the serotonergic system development in that particular period, if you are going to enhance the serotonergic system, it kills the synapses there. Later on, when they become adults, they exhibit the depressive-like symptoms, and those symptoms can be studied even in the laboratory animals. It may be lack of pleasure like anhedonia, or you have the learned helplessness, that's the behavioral despair. So these two major components associated with cognitive deficits can be studied in great detail. So that's what we study in the laboratory animals, and we did show that if you induce the endogenous depression, in the early phase of life, and when they become adulthood, they do have these the major components of the depression. And we assessed the various components of the learning and memory. It may be the working memory, or it may be special memories, or different tools we adopt, and we notice that there is a learning and memory deficit is seen in the major depression, which is caused by the clomipramine administration in the early phase of the serotonergic development. And more importantly, so you do have the anxiety-like behaviors, and that translates into the involvement of the other regions of the brain beyond the hippocampus and prefrontal cortex as well. What we get to see that, a large number of patients with major depressive disorders, we look into the structural and functional connectivities in the human brain. So how it gets altered. So in that perspective, it becomes extremely important how the structural changes in the different regions of the brain, specifically the prefrontal cortex, hippocampus, and amygdala, those three regions seems to be responsible for the major depression. So we need to understand what is the structural plasticity associated with it. So we looked into the hippocampal volumes, or it may be specifically looking into the dentate gyrus. That is the region where you have the antidepressant drugs. They act onto the dentate gyrus, and including the enhancing the neurotropic factors and also the formation of newer cells in the hippocampus that you get to see even in the chronic antidepressant treatments as well. So what we found that in the hippocampus as such, the volumes are not altered, but the dentate gyrus, the size of the dentate gyrus has significantly decreased. As I told you, the depression is also associated with the anxiety and also the mood disorders. And you have another brain structure which is called as the amygdala. The amygdalar volumes have been significantly increased. So as we know that we have the hippocampal functions decreases And parallelly, you also have the anxiety and the emotions and the fear responses. They get altered in the major depression. That is because of the involvement of the amygdala. So in the amygdala, the volumes has been significantly increased. This mimics the human conditions as well. Even in the major depressive disorders, when you do the MRIs and the structural and functional MRI, we notice that so you have the degeneration of the prefrontal cortex and also the volumes of the hippocampus, prefrontal cortex decreases, whereas the amygdalar volume significantly increases. So what is most important is that, when we want to treat the endogenous depression, and it has to be targeted to the dysfunctions, neurochemical dysfunctions, specifically the serotonergic system and noradrenergic systems, which are majorly involved in the major depression. So we observed that the serotonergic dysfunction has been, replicated in this animal model as well, and also the noradrenergic neurotransmission gets significantly altered. So all of these changes, structural and functional changes or the behavioral changes, has to be translated and understood in terms of the networks. So how does the networks and the information processing in the brain is altered in the major depression? So for that, what we do is, we do the electrophysiological studies. We take the live slices of the hippocampus and we stimulate certain pathways, and then we elicit the excitatory postsynaptic potentials, and we maintain them for a long period of time, and we call it as a long-term potentiation. So even for a brief period of the stimulus, and you have the enhanced activity in the hippocampus. So the hippocampus is a key brain structure which is crucially involved in learning and memory, specifically the declarative kind of memory, and also this is involved in the spatial map formations. So you have to know about the facts and events and also the places and faces. So the hippocampus is crucially involved, and it is responsible for translating the short-term memories to the long-term memories. So every time you are listening to me and the contents are reaching into the hippocampus, you have the enhanced activity which is happening in the hippocampus. So the enhanced activity of the hippocampal circuitry is maintained for a long period of time, and we call it as long-term potentiation, which is a basis for the formation of the memories or translating the short-term memories to the long-term memories as well. Look at here in the depression, the red one. So you do have the LTP-like behavior, but it is not sustained and it's not for a long period of time. It is very short period of time. That means the ability of the hippocampus to process the new information when the animal is learning or an individuals are learning, it gets significantly affected. We get to see that, and this is maintained for a long period of time as well. A lot of studies has been performed to look into what is the structural plasticity associated with the major depression in the hippocampus or prefrontal cortex or in the amygdala. A lot of studies has been performed by us, and also we had an opportunity to look into the postmortem brains also, some time back. And we did notice that in the major depressive patients, there is a loss of cells. In addition to the loss of cells, you also have the connections has been lost. So you have the button-like structures, they are called dendritic spines. These are the sites where the communication between one cell to other cell happens. But in the depressive patients, in the chronic major depressive patients, you get to see that the button-like structures or the dendritic spines has been significantly lost. So that clearly indicates that, at the different levels of neural organization, it may be at the behavioral changes or at the structural level or the biochemical level, even at the molecular level, you do have a significant impact of the depression, and we need to look into the modalities of treating. So what is very, very interesting is that, so the current treatment, what is available is the pharmacological management with antidepressant drugs, and we use them. And over a period of time, about 3 to 5 years or so, what you get to see is that drug resistance. So at that time, you are going to change the dosage and you are going to add more drugs into that one. So you have the anti-serotonergic drugs and SSRIs, and you are going to add SNRIs, and in combination of them. And you have the undesirable side effects are there. There is a recurrence and also multiple drug treatment and treatment-resistant depression. Among the population, what we get to see global scenario is that, including in the Indian population, about 30 to 40% of the depressive patients by 4 to 5 years time, they do develop the resistance to the antidepressant drugs. So it is very, very challenging. So what is the next therapy? So you have to do the RTMS. So you have the repeated transcranial magnetic stimulation, or you give the ECT when you are not able to manage. So it is in the major depressive disorders, and it will translate into the schizophrenic symptoms as such. So you have the electroconvulsive therapy is the major therapy which we are going to do it. So it is very, very challenging. So for that, we need to evolve a different strategies and to come look into the more effective therapies in a modifiable way. So what is very surprising for all of us that last 60 years, there is no new drugs has evolved for treating the depression. What we have done is we just modified, repurposed those drugs. That's all is the achievement we have done it. So that means there is a lot more we have to think in terms of understanding the pathophysiology associated with the depression, understand the physiology, and then try to see that whether the new therapies and the novel therapies can be emerged. So when we were looking into the patient's data, we get to see that the major depressive patients, when as long as they are in the hospital, their depressive symptoms are better managed. Once they go back home or to the workplace, because you have those stressors, which is very much there in the society and it's there in the families. So again, you have the recurrence of the symptoms you get to see, and it comes with a much more robust way. So repeated recurrence of the depressive symptoms will have a major impact onto the structural and functional integrity of the brain. So we need to evolve the novel strategies. Is it possible for us to take care of these patients, providing a good environment? In addition to that, is it possible for us to reduce the doses of the antidepressant drugs? So reason is that even in the rehabilitation, which we perform at NIMHANS and other places, is that as long as they are in a controlled environment and they are being well taken care, so their symptoms are better managed. But once they go back and the symptoms worsen, and also they have a repeated episodes of the depression, that is very bad. So for that, what we have done is it possible for us to have the positive environment and also have the reduce the doses of antidepressant drugs? For that, we mimicked the human condition of the physiotherapy or the various kinds of these psychiatric treatments which they perform, where you have the rehabilitative tools. You could have enhanced locomotor or physical activity, sensory stimulation, social stimulation, cognitive stimulation. So we call it as a rat version of the Disney World, and where these animals has to perform the different tasks and the rewards they receive, and that will have a positive impact onto the structural and functional plasticity of the brain. While these animals are subjected to, depressed animals are subjected to a short period of the enriched environment, so we are going to reduce the dose of the effective dose of the antidepressant drug to the 50%. So with a good environment, along with the antidepressant drugs, is it possible for us to achieve in terms of reducing the depression in these animals? So that we have studied in a very great detail, and we did see that the anhedonia, learned helplessness, behavioral despair, all those symptoms can be reduced, and also the learning and memory, which is associated with the depression, the deficits can also be restored with a brief period of enriched environment, and in addition to that, you have the antidepressant drugs. So we studied all these things in a greater detail, what the structural and functional plasticity associated with that, and we provided the evidences, and which has transformed and translated into the newer approaches for taking care of the major depression. So you have multiple things as translated. And more importantly, even at the synaptic plasticity or at the networks level, where the information is getting processed, even in the hippocampal synaptic plasticity could be restored when you have the combinational therapy or when you have the synergic action of the good environment along with antidepressant drugs. So we did that one, not only the SSRI, we also studied that in great detail with SNRI. Does the SNRI also have the same effects? So we did study the serotonergic reuptake inhibitors and also the noradrenergic reuptake inhibitors also, looking into the mechanisms, how does it going to restore the abnormal synaptic plasticity in the hippocampus and the other regions of the brain. So more importantly, what is the take-home message from all these studies which has been done for several, more than 2 to 3 decades, is more than 30 years, is that, so it's not only the drug alone, so you need to provide a conducive environment when it comes to the mental health issues. It's extremely important. So your family, the whole family has to support when it comes to the mental health disorders. And also in the workplace, so you need to have all of the different systems. Now you have in the schools and colleges, you have some counselors. So now in the universities, it has become mandatory that you need to have the stress management. For that, you have the counselors are very much there. So until unless you have a multi-pronged approaches to handle a complex disorders of brain dysfunctions, it is very, very challenging. Okay. So now more importantly, we could also use different kinds of the tools where you could stimulate certain regions of the brain, which is very, very beneficial. So you can have the non-invasive, repetitive transcranial magnetic stimulation. You need not push all the patients until unless it is necessary in the manic disorder or in the major depressive disorders. Only the ECT has been advocated. So that with a modified ECT, we do it. So we try to understand, is it possible for us to stimulate the certain regions of the brain, which is going to be beneficial in terms of reducing the symptoms associated with the major depression. So I'll not go into the details of each one of them, and I'll just give the summary what we have studied from the want of time, and we are already late. Okay, so what did we do it? So we implanted the electrodes into the brain, and then we stimulated the certain regions of the brain, specifically the reward regions, and it had a positive impact on reducing the depression. It reduced the depressive symptoms and also the associated cognitive declines, and it is also could alter the microenvironment of the neurons in the hippocampus, prefrontal cortex, and in the amygdala. So we looked into how does the neurons and the glial cells they do interact, and what are the downstream signaling molecules and the molecules which are responsible for neurodegeneration and also the recovery process, including looking into the trophic factors and also the neurogenesis. So several components has been studied. So that means if you could have a specific stimulation of the certain regions of the brain, and that will have a huge impact on reducing the depressive symptoms. In addition to that, if you could have a preventive role as well, if you know that the certain regions of the brain, it can be enriched so that the resilience for handling the stressful actions or stressful effects or driving the stress things into the depression can also be minimized. So all this is possible because the brain is so plastic. So that gives a greater opportunity for neuroscientists for us to understand. You have the regressive plasticity and the progressive plasticity. So every actions what we do and our behaviors are modulated tightly with various factors which are responsible for altering our neural circuitries, which will have a positive impact that we call it as a positive progressive plasticity, or you also have in the various disease conditions, you call it as a regressive plasticity. So that means clearly there is a continuous rewiring and remodeling of the brain circuits which occurs throughout our life, even before birth till the end of our life, which is responsible for the altering the neural circuitries, and that can be better managed with various kinds of the tools we are going to use it. So what is missing? So what is that which is going to bother us? Nuclear families, there's no social interaction, there's no social well-being. So each one of those factors, so it's the family structures. So the humans are social animals. Our sociability has significantly decreased, and sharing of information has been through your WhatsApp or phones. So not with the physical contact with, you never go and meet the people. So greet the people and interact with them. So share the information. So all those things which has reduced your sociability in the society and in your community and in the family. And these are the examples, and we drive into that situation. So I want to thank my research group, the depression research group, and thank you very much for all the collaborators and the funding agencies. So thank you very much. Dhanyawad.
Thank you, sir. It was very interesting talk. It's not only about drugs, but also you have stressed on the enriched environment. That is very important because it is involving the productive age, 20 to 40. So very enlightening lecture, sir. Questions we can take.
If you have any questions, and you can ask, we have only 1 minute. Any two burning questions? We're running out of the time, so I have to also run.
Actually, it was a very interesting session, sir. As a clinician, yes, I have seen patients these days going into depression a lot, especially in diabetics, and hypoglycemia adds to this, wherein they go on, especially when patients come to us with hypoglycemia, and I have seen a early development of dementia in these patients. Say around 60 patients starts developing dementia. Maybe there is some amount of this thing in
In fact, in collaboration with my teacher, we did study that in great detail. So how does the diabetes affects the learning and memory, and also is the hippocampus and prefrontal cortex is susceptible for that. We did see that, okay, the diabetic individuals are more susceptible for the depressive like symptoms, including anxiety.
The question is this reversible?
Mm.
And what
The-
time frame are we looking at?
No, it's extremely important that, what is the treatment regimes, how soon you are going to treat, how continuously you monitor them. So most of the time it is challenging as a clinician so that, you treat them, but subsequently the follow-ups is not done. So even the patients, they will stay away. So until unless there is a close monitoring which you are going to do it, and it's going to be very, very challenging. But you do have the relation between diabetes and the mental dysfunctions.
Do you see a role for melatonin in this?
Yes, certainly yes.
Because there is a lot of news about, we are told that melatonin may be the antagonist of many of the problems which cause depression. So have you
It is
explored that?
associated. It's associated. We did a biorhythms and also look into another structure of the brain. It's called as the subiculum. The subiculum is the key factor which is responsible for regulating our biorhythms and including the sleep patterns. We did study in great detail, and we noticed that the melatonin changes and also the biological rhythms will have a significant impact on the functioning of the neural circuitries, including the cognitive functions.
Any questions from the audience? Yes. No, if that's so, I think we should conclude here.
So now we request the chairpersons to present the certificate of appreciations and a memento to our speaker. Now I request Dr. Aruna Bhavani ma'am to come on dais and hand over certificates to our chairperson. Thank you, everyone. Now we shall go for break lunch, which is arranged at the basement.
Yeah.
And we all will reassemble here at 2:30 for further session.
Yeah, so just a small break before you all go to lunch. The association has a journal called Indian Journal of Physiology and Pharmacology. And as scientists, all of us, we require publication for many purposes, and these days largely for our CV. So good journal benefits because of publication. The journal gets high impact factor value, everything. The authors get publications, they get in the CV. But there's one group of people who work behind who are never recognized. And these are the peer reviews. And APP has begun this activity to recognize the peer reviews, because without them, neither the authors benefit nor the journal. And so we started this thing of that we take the whole year's data, and those of us who have been contributing to peer reviewing. So we're recognizing 3 people from pharmacology side and 3 from the physiology side. And for this year, we have these Dr. Kiran Prakash, Dr. Kumar Sarvottam, Dr. Priyanka, Dr. Ankul, Dr. Lalit, and Dr. Vijay Kumar. May I request Professor Machado and Professor Shashidhara, we request your presence to encourage our peer reviews. And I also then request Kiran Prakash and Vijay Kumar to come up and receive the award for the same. Both of you come together, please. Yeah, please. Right. Peer reviewing is a very painstaking job. It takes hours, perhaps sometimes more than the authors themselves may have read the paper. And we are very thankful to Kiran as well as Vijay. So it's a
Association of Physiology and Pharmacology. So you take together and we hope that this is a motivation to rest of us to participate in the process and review journals as fast as possible. Vijay. Thank you very much. Thank you very much, both of you and everyone. And thank you, Professor Sasidhara and Professor Machado to do the honors for us. Thank you very much, sir. Oh, thank you.
Thank you, everyone. This is the time for lunch break, which is arranged in the basement hall. And you please all assemble here at 2:30 for further sessions. Thank you.
Screen sharing. ये शेयर हो रहा है कि नहीं हो रहा कैसे पता चलेगा?
Recording in progress.
Well,
Light lock போடுறேன்.
Hello. Hello.
Next is yours,
From 2:30 onwards. is it?
from 2:30 onwards. Okay, then he might be having.
Huh?
Then she might be having and she might have downloaded. I'll ask the person. I know. On what topic, sir? Sorry.
I got some.
Hello. Hello. Hello, am I audible? Hello.
Hello. Hello.
Hello.
Hello, good afternoon, sir.
Yeah.
It's actually a lunch time, so people are coming back. So
Okay, okay.
can you please just for five,
Sure.
10 minutes?
Sure, sure,
The
ma'am.
audience will come back and we can join then.
Sure.
Thank you.
ಈಗ ತಗೊಳವ
Next person is Dr. Rohit Sinha, sir. He is online and he is going to present for the topic Resmetirom for NASH from concept to cure by Rohit Sinha, sir.
Thank you very much. At the outset, I wish to thank the organizing committee for giving me an opportunity to share this topic with the people here. And I'm sorry I could not come here physically for the conference, but I hope so I'll be able to share some of these interesting findings on which I was a part of it, and this drug which was recently approved for treatment of NASH last year. So I'll share the screen. Yeah, I hope it's visible. The screen, is it?
Yes, sir, it's visible.
Yeah. Okay. So resmetirom for NASH is a topic is because it's very important for both physiologists and pharmacologists here, is because it basically covers a journey of a drug from a hypothesis step to preclinical stages, and finally how it made up to the FDA approval last year for this treatment of NASH, for which there was no drug approved before that. So just to give you a brief background of how a drug development process take place. So it starts with basic science research, which is basically the hypothesis building stage of any drug development, which is followed by preclinical trials, which is done in animals and in non-human primates, and followed by phase 1, phase 2, and phase 3 clinical trials. And finally, if everything is fine with the phase 3 clinical trial, it is taken up for consideration by the FDA for approval as a drug for a treatment. So it's quite a lengthy process, and the whole process takes many years to get approved. And this was the case for resmetirom also. And I was basically involved in the preclinical testing of thyroid hormone as the concept building step of whether thyroid hormone can be used as a treatment for non-alcoholic steatohepatitis. And just to give you a background of what this disease is it's becoming a major epidemic in the world, and especially in our region. And this NASH, which is known as the non-alcoholic steatohepatitis, is basically classically defined as liver disease in which inflammation can be seen, and this is seen in the patients who don't drink a lot of alcohol. So it is basically made to differentiate from alcoholic steatohepatitis and followed by cirrhosis. So now the terminology has changed a bit. So the new guidelines rename NASH as MASH, which is metabolic dysfunction associated steatohepatitis. And this is because most of the patients with NASH and the umbrella term, which is known as NAFLD, which is non-alcoholic fatty liver disease, are basically also have other metabolic disorders like diabetes and hypertension. So it's basically a metabolic driven hepatic pathology which is seen here. So if you can see in the slide is that a normal liver, you can see hepatocytes which are you can see a healthy hepatocyte with a little bit of lipid droplet. But once you progress to this NASH stage, what you can see is that there are a lot of fat accumulation in the hepatocytes, which is also followed by cell damage, infiltration of neutrophils, and appearance of fibrosed tissue between the cells. So this stage is basically known as NASH, and it is preceded by a stage which is known as benign steatosis. So that is basically just a buildup of fat, and it is not clinically relevant. Only when it progresses to inflammation stage, and when your liver function test starts showing up, your ALT, AST goes up, it is where it is a clinically relevant stage for intervention. And NASH then in many patients progress to cirrhosis and hepatocellular carcinoma. So it's a very serious problem nowadays. And this is basically driven by a lot of lifestyle related changes which are happening in the industrialized world. and basically high fat, high calorie diet and physical inactivity are the major driver for these disease. And in some cases there are also some genetic determinants which also basically predispose you to develop NASH. So if you see its prevalence in the world, it's quite a serious problem in Indian subcontinent, as you can see in this slide. That at present is an estimate that we have 33.83% of population affected by MASLD. So MASLD is the umbrella term on which some of the patients do develop MASH or NASH. So it starts from benign steatosis to cirrhotic stage. And therefore, it is a big health crisis in most of the parts of the world. And there is a urge to develop drugs for this disease. So before 2024, the only way to cope up with disease was to change the lifestyle, do exercise, eat less of high calorie diet. And this was the only thing a doctor could tell the patients. But it's good that now we are coming up with new drugs to treat this disease also. Now, if you see the link between thyroid hormone and NAFLD. So, the first clinical studies which started showing up were in 2009 and 11, which showed that indeed hypothyroid patients which have low circulating thyroid hormone level were more likely to develop a non-alcoholic fatty liver disease, and many of the patients do develop non-alcoholic steatohepatitis also. So this was indication that possibly thyroid hormone have some role in the development of NAFLD and NASH. So a lack of these hormone, either circulating hormones or deficiency of thyroid hormone action within the liver itself could be a driving force for NASH development. And there was a study which showed that in fact in advanced fibrosis stages, like F3 and F4 stages, there were reports of increased expression of few enzymes which regulate the metabolism of thyroid hormone in our liver. So this is a healthy liver, a mild fibrosis stage and advanced fibrosis stage in human NASH patients. And what was seen was that DIO1, deiodinase 1, which is an enzyme which produces active thyroid hormone in the liver, by converting thyroxine to triiodothyronine, was significantly downregulated in NASH liver. And the other enzyme DIO3, which is an enzyme which inactivates thyroid hormone. So this enzyme basically reduces the ability of T3 to bind to its receptors and activate cellular action, was actually increased. So not only the systemic circulation of thyroid hormone, which was decrease in hypothyroid patients predisposed them to develop NASH, but also in NASH there was a picture of what you could see, just like you see insulin resistance in liver cells in diabetics. We were also seeing decrease thyroid hormone action in the liver. So there was a type of thyroid hormone resistance ectopically developed within the liver cells. So now the question is, can we increase thyroid hormone action in the liver as a way to inhibit or reverse the NASH phenotype, which is seen? So just to give you a brief background of how thyroid hormone actually work, is this slide basically summarizes how thyroid hormone actually work in any of the cells, whether it be hepatocyte or cardiomyocyte. And so it starts from the release of thyroid hormone in form of T4 and T3 from the thyroid gland. It enters the cells through specific thyroid hormone receptors. And when after entering inside the cells, it is converted by deiodinase 1 and 2 into active form, which is called T3 or triiodothyronine. And it is also inactivated by DIO3 into reverse T3 form, which is not biologically active. And after entering, the T3 itself can either activate mitochondria action by non-genomic action, but in the most of the cases, T3 is a ligand for a nuclear receptor in the nucleus, where it binds to these nuclear receptor known as thyroid hormone receptor alpha or beta. So there are two isoforms of thyroid hormone receptor. which in turn regulate the gene expressions of several genes regulating metabolic functions or growth functions. So this is how at a cellular level thyroid hormone acts. So now the question was that how thyroid hormone regulates lipid metabolism in the liver cells. So because if hypothyroid patients are developing more fatty liver, then definitely there is some role of T3 in regulating the lipid inside the hepatocytes. So what my initial work during my postdoc period was to understand how thyroid hormone affects lipid mobilization inside the hepatocytes. So what we found was that thyroid hormone when injected into animals. So these are C57 black mice which were injected with T3. And what we found was that first of all, thyroid hormone treated mice liver had increased number of mitochondria, which is shown here as with arrowheads. So definitely it increased the oxidative capacity of a hepatocyte to burn fat. But at the same time, it increases the mobilization of lipids shown here with the arrow into structures known as lysosomes. So these are basically degradative structures which degrade lipids because they have lysosomal lipase inside them. So what we found was that there was an increased autophagy mediated degradation. So autophagy, basically what happens is that we showed that these structures known as autophagosomes uh, are basically fusing with the lipid droplets. So you can see a big lipid droplet here, and these autophagosomes fuse with lipid droplets and bring them inside the lysosome. So what you can see here is the autolysosome, which is basically a lysosome containing all the lipid droplets inside the hepatocytes. And this is how our thyroid hormone activate this process and helps the liver cells to burn fat. And that is why possibly when you have low circulating thyroid hormone, even at a systemic level or at the intrahepatic level, you do accumulate a lot of lipid in the hepatocytes. Now, the question was, if we inhibit this process of autophagy or lysosomal mediated lipid degradation, whether it affects mitochondrial mediated lipid oxidation. So we know that lipid can be degraded by beta oxidation, which happens inside the mitochondria. So what we did was we used siRNA. So it's a genetic approach to block the activity of any gene. And in this case, we basically inhibited the expression of ATG5, which is the autophagy regulating genes. So when you don't have ATG5 inside the cells, you basically don't activate autophagy. And because you don't act autophagy, what we predicted was that you should not have increased lipid oxidation because the fat is not being shuttled into the into the lysosome, which is then degraded into free fats and can get inside the mitochondria. So indeed, this was the case that when we gave T3 to animals, it increased the beta hydroxate hydroxybutyrate level, which is a basically ketone bodies which are produced as a result of fat oxidation. But when we inhibited ATG5, so when we blocked the autophagy, mitochondria inside the hepatocytes were no longer able to burn the fat. So this is what the model looks like from our study, was that T3 basically activates the degradation of triglyceride inside the lysosomes, which release free fatty acid in the cytosols to be taken up by CPT1 alpha. It's a protein which basically takes free fatty acid inside the mitochondria for beta oxidation. So this is how we were one of the first to establish that thyroid hormone or thyroid hormone analogs could actually be used to increase beta oxidation of fat inside the liver cells by activating this process known as autophagy or lipophagy. Now, what about a disease model, preclinical model of NASH? So, in this model, what was seen was that in parallel to increasing autophagy, these model actually showed decrease in NASH score, overall NASH score, which is shown here as steatosis, ballooning, and inflammation. So you can see here that in these mice, these were the control mice which were given normal chow diet for 24 weeks. NASH inducing diet was basically a diet in which Western diet, which is basically high in fat and carbohydrate, was given for 24 weeks, and thyroid hormone treated group was basically mice fed NASH diet till 16 week, and the rest 8 weeks they were given a combination of T3 and T4. And then the assessment was done in terms of liver histology. So what we were able to show is that that thyroid hormone induced autophagy is a way in which we can basically burn fat and also decrease inflammation and other cytotoxicity effect of fats inside the liver cells. Now, this was a proof of concept preclinical studies done in animals to show that thyroid hormone can indeed be used as an anti-NASH agent. And to go to the next step of drug development stage, you must show that in preclinical, in clinical trials in humans also this concepts hold. So what we did was a phase 2 clinical trial in Singapore. What we did was we gave thyroxine to patients. So thyroxine, basically what we had to do is that we have to give them T4 such that they don't become hyperthyroid. So in their T4 was reduced in a way that they were in the bottom quartile of the TSH level. They were not hyperthyroid, but the T4 levels were certainly increased to see the effect on liver fat levels. So these were basically patients with type 2 diabetes with steatosis. So what we were able to show was that T4 administration in patients were able to decrease intrahepatic lipid levels. And in patients above 50 years of age, they were also helpful in reducing the HbA1c level, which is a parameter of insulin resistance. So this was a proof of concept to show that thyroid hormone can in fact be used as a possible treatment for NAFLD, and thyroid hormone analogs could be the next step for taking up as a therapeutic regimen for treatment of NASH. So now the problem in pharmacological area of using thyroid hormone analogs as a treatment for this has been around for a lot of time, for quite a lot of time for treating triglyceride hypertriglyceridemia and obesity. So the only problem with using thyroid hormone as such in a high dose or a pan-thyromimetics, means thyroid hormone analogs which can target multiple organ, was that we have adverse effect in terms of cardiomyopathy in the heart and excessive bone loss in the skeletal system. Other than that, the other problem with using natural thyroid hormone is that you can actually suppress the pituitary thyroid axis, HPT axis. So when you give thyroid hormone exogenously, you can actually inhibit the endogenous production of thyroid hormone. So this is basically the reason that many of the earlier thyromimetics failed in phase 2 clinical trials, because they were having adverse effect on heart and bone. So the reason is that different tissues in our body have different thyroid hormone receptor. For example, the liver is basically enriched in the thyroid hormone receptor beta, whereas bone and heart express basically thyroid hormone receptor alpha. So if you want to have effect of thyroid hormone in the liver and sparing the heart and the bone, you should have a thyroid hormone analog which can only be activated in the liver and doesn't bind to receptor alpha, which is enriched in bones and heart. So this was basically overcome by pharma companies, especially Madrigal, which was pioneer in making resveratrol. So this is basically a modification of the natural thyroid hormone T3. But the beauty of this analog is that it only passes through a specific thyroid hormone transporters known as OATP-B1. So, and this transporter is only enriched in the hepatocytes. So it won't enter any other cell other than the hepatocyte. Plus, it only activates the thyroid hormone receptor beta and not the alpha form. So what this slide shows you is that this is a in vitro study to show the specificity of MGL3192, which is a chemical name for resveratrol. Just a second. Yeah. So these basically are the HEK cells which are overexpressed with OATP-B1 transporter, which is a thyroid hormone specific transporter expressed in hepatocyte. and it was transfected with a luciferase gene with under a response element for thyroid hormone receptors. So what the concept is that if you add MGL 3196, which is resveratrol, and you can show that it only activates through thyroid hormone receptor beta, and it only activates when this OATP B1 is present. That shows you the specificity of this analog. So this luciferase activity is basically gene activity, which shows that when you add T3, so these triangles, red triangles, and this round triangle is basically T3 added to cells expressing thyroid hormone receptor or beta. And you can see that natural thyroid hormone basically activates thyroid hormone responsive gene in a similar way. So they bind with a similar efficacy to both thyroid hormone receptor and beta. But when you add MGL 3196 in expressing thyroid hormone receptor alpha, you don't see an increase in gene expression, luciferase expression. Whereas when you have MGL 3196 added to cells transfected with thyroid hormone receptor beta, you have almost 90% efficiency compared to the natural thyroid hormone levels. So this shows a specificity for this analog, thyroid hormone analog. And when you transfect with another homolog of this transporter OATP B3, you don't see, you still see a leakiness of expression with MG112, but it's not as good as when you see with overexpression of OATP B1 transporter. So this showed that resveratrol indeed is a very specific analog for hepatocytes, and it only activates thyroid hormone receptor beta receptor, which is enriched in the liver and should not have any adverse effect on heart and bone. So finally, with these positive results, phase 2 clinical trial was done with resveratrol in patients with NAFLD, and the results were quite encouraging. So they were given resveratrol, and absolute fat reduction was analyzed in the liver at 12 weeks and 36 weeks. The study was published in Lancet in 2019. And what the study showed was that there was a significant reduction in liver fat levels in given at 12 weeks or 36 week at both 60 mg and 80 mg doses of resveratrol compared to the placebo controls. And other secondary outcomes were also quite good, like it reduced the LDL cholesterol, triglyceride levels, and also were helpful in reducing the ALT levels at week 12 and 36 week. So following these encouraging results by phase 2 clinical trial, last year the phase 3 clinical trial also was completed for resveratrol, and this was published in NEJM and showed a really good result, not only in reducing liver fat, but also the more clinically relevant areas like worsening of fibrosis. So basically, resveratrol was able to basically reverse the fibrosis, initial stages of fibrosis in patient with NASH, and this was significant at both 80 mg and 100 mg doses of resveratrol. And this was also followed by beneficial effect on other secondary parameters like LDL cholesterol and triglyceride levels, and also liver function tests were observed to be improved in patients receiving resveratrol compared to placebos. So finally, with these positive results, last year the FDA approved resveratrol as the first pharmacological drug for NASH, and this was a very happy moment for all of us, including me, who have been involved in this study of using thyroid hormone or thyroid hormone analogs for treatment of non-alcoholic fatty liver disease and NASH. So, but still, there's a long way to go, and there are some challenges and future directions which are need to be kept in mind. And this is the long-term safety data is still not out here. So, this will basically the phase 4 studies which will take some time to see what are the adverse effect. In the short term phase 2 clinical trials and phase 3, the major side effect was basically diarrhea, and that too also were observed to be insignificant after long-term usage of resmetirom. So we still don't know a long-term adverse effect, but at present the drug looks pretty safe. And we still don't know how long we need to treat the patient to see the effect of resmetirom, so that no reversal of NASH can happen. And we still don't have data. The clinical data for in patients with lean NASH is not out there. So NASH can happen in both obese people and in lean people. So the most of the studies done in the West were focused on patients who have obesity and diabetes along with non-alcoholic fatty liver disease. But the studies had not been conducted very well in especially Asian countries where we have lean people having fatty liver and NASH. So these are some areas in which more study have to be done. And effect of non-hepatic outcomes and complications during the phase 3 and phase 2 clinical trials, it was not observed that they had major side effects in terms of effect on heart and skeletal muscle, and data on survival benefit is also limiting at present. So with that, thank you very much, and open for discussion.
Thank you so much, sir, for this wonderful session.
Thank you.
Any questions?
Good afternoon, sir. Basically, I am from CFA. I am professor of physiology. I have been working in physiology department since 2006 in CFA. Recently, your research is on liver fibrosis. So one of my known person was admitted in hospital for gallbladder stone. She was admitted, surgery people accepted her, and according to the biochemical analysis, her thyroid level was normal, but she had liver fibrosis. But that is not coinciding with this thing, your fact. You already in your whole lecture you mentioned about liver fibrosis, and having patient will have less than normal thyroid level.
Yes.
In this case, her thyroid level was normal, but still she was suffering from fibrosis. And then Dr. Kanhaiya is there, gastroenterologist. He operated this patient after consultation with Dr. Kanhaiya. I do not know whether Dr. Dr. Kanhaiya or not. He is gastroenterologist. He operated this patient, and now she is totally all right.
Yeah.
So in physiological point of view, actually liver fibrosis, it is reversible process.
Yes.
I think as per your information, she was not taking regular physical exercise, so that may be the reason why she was.
Yeah. Definitely not all people will have a thyroid dysfunction to have a NASH.
Correct.
So basically, the studies we show is that if you have hypothyroidism, you should go for a regular check for your liver, because it predisposes you to having NASH. But it is not that all patients who have NASH will have a thyroid dysfunction. So that is the main.
Means it is not compulsory that person.
It is not compulsory. Definitely, there are many more reasons to have a liver fibrosis and NASH.
Thank you, Dr. Thank you.
Thank you so much, sir, for joining us, not offline, but online. Whatever you said and teach us, it's really worth. Thank you.
Thank you.
Just please be seated for 5 minutes. I'm going to start just in a minute.
For our next session, I kindly request Dr. Ashok Kumar Jarial, Professor of Department of Physiology in AIMS Delhi, to kindly chair the session. I kindly request, sir, to introduce our next resource person on the talk Hypertension Care Cascade Among Native Rural and Tea Garden Population Assam by Dr. Prashant Kumar Bora.
Good afternoon. It's my pleasure to introduce Dr. Prashant Kumar Bora, a scientist F, ICMR, in the Northeast region of Dibrugarh. And we're going to speak on a very interesting title topic, a hypertension care cascade amongst native rural and tea garden population of Assam. Many of us have sort of believing that the green tea and the other teas are helpful for lifestyle disorders. And so it'll be interesting to see what Dr. Prashant Kumar Bora is going to share with us. May I request you, sir, to begin your talk, sir.
PPT was already downloaded. I share to Dr. Arpit. I did.
Respected all, a very good afternoon to you. At the very outset, I like to offer my sincere thanks to the respected chairpersons, organizers for giving me this opportunity to be here and to present my works. Thank you once again. So my topic is hypertension care cascade pathway among the native rural and Tibetan population of Assam. So as you know, the cardiovascular disease are the major causes of illness and disease burden. And these are responsible for approximately 393 million disability adjusted life years as per 2019 data. It is the main cause of death and disability worldwide, accounting for around 18.6 million deaths. And raised blood pressures or hypertension is the prominent risk factors for cardiovascular diseases. And if you see the global prevalence, it is around 31.1%, and Indian prevalence is around 31 to 35.5%. And it contributes to around 235 million dailies and around 10.8 million deaths. And approximately 88% deaths are in low and lower middle income countries. So as you know that there are effective treatments for hypertension exists, and it is cost effective also, but there are significant gaps in terms of hypertension awareness, treatment, and control, which may vary across the regions, across different communities. Factors associated with poor hypertension controls are to be identified. And health system also requires transformations in a resource-poor settings. And understanding this hypertension care cascade at the community level will help better management of hypertension. So we aim in the study to find out the prevalence and risk factors of hypertension, to identify the hypertension care cascade pathways among the native rural and the Tibetan communities of Dibrugarh district Assams, and to identify the predictors of different elements of hypertension care cascade. So coming to the methodology. So main data source is the health and demographic surveillance systems, which was carried out in our centers that covered 60 villages and 20 tea gardens, and we included 1,06,769 populations. So this is the site. Actually, we took ICMR RMRC Dibrugarh Center, the center point, and we included all the consecutive villages and tea gardens. So this is our sampling strategy. So the total population is 1,06,769. We excluded participants below 18 years, that is around 33,822. And we also excluded pregnant women, participants with chronic debilitating diseases, major psychiatric diseases, and missing data, which are very much required for the analysis of the present communications. And ultimately, ultimate sample size is 53,301, which is around 50% of the total individuals of the Dibrugarh SDSS. So for the data collection, we trained staff, they collected the data using one in-house mobile applications, which was developed by us. We measured the height, weight, waist-hip ratio. Blood pressure was monitored using Omron digital blood pressure monitors. We took 3 readings, and the mean of the last 2 readings were considered for the analysis. And these are the variables we collected from the demographics, behavioral variables, and we use these definitions. Blood pressure, high blood pressure was categorized using European Society of Cardiology, European Society of Hypertension Guidelines 2023. So for in simple words, hypertensions, systolic blood pressures greater than or equal 140 millimeter mercury, and or diastolic blood pressure greater than or equal 90 millimeter mercury, or if the person is on antihypertensive medication. And we consider any participant under control blood pressures if the blood pressures after medication is under 140 by 90 millimeter mercury. So for defining the hypertension care cascade, we use these definitions. So total hypertension, that is 140 by 90, and or antihypertensive medications. On-screen means blood pressure 140 by 90, but they never had measured or never diagnosed. Screen but undiagnosed, that means they are the unaware. The blood pressure was measured by some other ailments, and the participant is also not aware of that. Diagnosed and aware, but not medications. So this group of participants, they are aware, but they are not taking the antihypertensive medications. Diagnosed and aware, that means aware, and they are on medications. Again, there are two groups, uncontrolled and controls. So for the statistical point of view, we use chi-square test for categorical variable, and we also carried out binary logistic regression analysis to find out the risk factors of hypertension as well as the independent predictors associated with different elements of the hypertension care cascade. Coming to the results, so total participant we covered 53,301. From the native rural community, 23,947. Tea garden community is this much. The majority of the participants were from the age group 18 to 30 years. Overall prevalence of hypertension is 27.3%, little bit higher among native rural population than the tea garden communities, but this is statistically insignificant. Again, male had higher prevalence of hypertension, 29.6% versus 25.4%, which is statistically significant. And in logistic regression analysis, we found that male genders, older age, illiteracy, widowed or separated, homemakers or business occupations, high BMI, low physical activity, consumption of alcohol, tobacco, and extra salts are the independent predictors for the hypertension. So this is the distribution of mean systolic and diastolic blood pressures in the whole populations. So you see that there is the incremental increase in the both systolic and diastolic blood pressures according with increment in the age groups. The same pattern we observe in tea garden community also. And again, same pattern among native rural community also. So coming to the prevalence of hypertensions, if we compare according to genders, among males, it is 29.6% versus 25.4% among females. For the native rural, if we compare between native rural community and the tea garden community, it is almost same. There is no statistically significant difference. Again, for the tea garden community, if we compare, always males show the higher prevalence of hypertension for the native rural population as well as tea garden communities. Now these are the risk factors which we identified. Gender, the male genders is the risk factors, older age, then lower educational levels, widowed and separated, business, history of diabetes, alcohol, tobacco consumptions, and consumption of extra salt or salt as a side dish, obesity, and tea-garden communities are the risk factors for hypertension. Now coming to the hypertension care cascade. So the total hypertensive we found is 14,562. So you see there are a reasonably higher numbers of the participants are screened, but the awareness is very less. It's only around 32.8% only for native rural and 25.8% for the Tibetan community only. And again, if we come to the treatment part, it is further coming down. And if we come to that control out of the participant taking the antihypertensive medications, it is very less, only 5.5% among Tibetan community, and it is around 9.3% for the native rural populations. So in different subgroups, the same pattern exists, but one important thing is that the hypertension care cascade shows favorable figures in case of females than in case of male participants. So same pattern, so I don't want to repeat. So the summary is that in the overall study populations, although screening is reasonably high, ranging 78 to 81%, A1S treatment and control are very low. Situation is far worse in Tibetan community than native rural populations. In the overall population, when analyzed across the genders, females shows favorable care cascade than males. And if we analyze according to the community, it shows that reasonably higher screening rate, but screening is reasonably higher, but again, same awareness, treatment, and control are very low. And when analyzed according to the genders, female shows favorable care cascade than the males. Now coming to the risk factors. So we have carried out binary logistic regressions to find out the independent predictors for different care cascade pathways. These are, in summary, these are the risk factors: younger age, low level of educations, students and unemployed participants. Marital status is also important. Normal weight, they usually ignores. Tea-garden workers, heavy physical activities, and alcohol consumers. So the pattern is almost same with some difference across different categories of hypertension care cascade pathways. So in conclusions, hypertension is a major cardiovascular risk. Expanding effective screening, treatment, and control is essential for better management of the hypertension. Community awareness, compliance to antihypertensive medications, accessibility to healthcare for regular blood pressure measurements, sustained antihypertensive drug supply are the need of the hour. And given the high prevalence and the large treatment gaps, multi-level interventions in respect to patients, provider, and the systems, prioritizing hypertension management is required. The strength, the sample size is very large, 53,000, and all the data were collected by trained investigators. But there is limitation because we measure the blood pressure in a single point of study because it was a cross-sectional study, and there might be some recall bias of the self-reported treatment history and other variables. So there are some other works which are ongoing in our centers. We are doing epidemiology of hypertension, salt sensitivity profiling, and we have developed an intervention module And we have also developed one Android-based mobile application with decision support systems for community control of the hypertension. We are also working on task shifting models with involvement of self-help group members for the community control of hypertension. So thank you.
I may start because it's a very great amount of work that you've done. So screening 1 lakh people is not an easy job. So, but your data on about 30% prevalence
Yes.
in a rural population, and our general thinking of people is that the rural population is much better in cardiovascular disorders as compared to the urban population.
Yeah.
How does that hold?
Actually, maybe 20 years back, the prevalence of hypertension among native rural population is around 33%. Now it is little bit decreasing. But for the tea garden communities, again 20 years back, the prevalence was around 60.8%, very high. Specifically for the tea garden community, they used to have the habit of taking extra salt. Very high amount of extra salt, maybe more than 15 to 20 grams. Although WHO recommends that only 5 grams should be there. They also have the habit of taking salted black tea at the workplace and at home also. We have done one intervention module targeting dietary salt restriction strategy. That was a single point agenda intervention modules. After that, providing salted tea at the workplace was almost abolished. That practice was almost abolished. So I think for that reasons, the blood pressure is coming
The
down.
salt was the major issue earlier. Now that is taken care from 60, it's become 30. That's what you're.
Sure.
I mean, earlier the salt intake was very high.
Very high, yeah.
And now that has been taken care, it's come down to 30%.
Yes, yes.
But again, looking from the sedentary lifestyle perspective of the urban life, the village life is thought to be better for health, but 30% still is a higher number. What would you?
Actually, the tea garden community, they are basically daily wage workers involved in the tea industry. And if you see the BMI pattern, they are lean and thin. So the major risk factor is the salt consumption only.
That's very
Yeah.
valuable lesson.
Yeah.
That salt is a very big factor.
Very big
And
factors.
especially in the urban setting, you may be looking lean and thin.
Yeah.
But still, you may be having hypertension.
Yeah.
Simply because of high salt intake.
Yeah.
Thank you very much for that valuable lesson to all of us. If there are any query from the house, please.
Well, very nice presentation. Thank you. I am really impressed by the numbers of the population in the rural area that present hypertension. It seems similar to the urban population, right? So this is bring for the discussion something like we expected that the rural population should be more active in terms of physical activity because they are working on the fields and less stress than the environment of urban large cities. So apparently the genetical backgrounds combined with the salt ingestion and other nutritional aspects make the difference in terms of hypertension. This, the impact of your data in terms of overall understanding of hypertension is really, really important because with this, as you mentioned, the large number of 50,000
populations,
people that were evaluated is amazing. Congratulations. And this will bring a lot of discussion for the fields. Yeah.
Yes, sir. Sir, to add your comment, actually, we have carried out one salt sensitivity phenotype study also. So we assess that whether salt increases the blood pressure. Salt means dietary salt. So we found that these peoples, the Tikaran community, Assamese, and we have carried out that study among Mijus also. So around 30 to 40% they are salt sensitive. That means when we introduce salt, it increases the blood pressure. So that's why that is also another reasons, uh, and for which we can say that yes, salt is a risk factors.
Yeah,
Thank you, sir.
Because the data that is to read in Harrison's spectrum medicine, there are two kinds of groups: salt sensitive and salt insensitive.
Yes.
There are some people who can take as much salt as they want, nothing happens to them.
Yes, sir.
And there is one group which takes salt, BP just shoots up.
Yes, sir.
So then this is that 30 to 40% group.
Yes, sir. We have done that study.
Yes. Thank you very much, sir.
Thank you, sir.
I request the chairpersons to present the certificate of appreciation and a memento to our resource person. Moving ahead with the talk on the topic, activating adiponectin receptor as a potential pharmacotherapy for mitigating osteosarcopenia by Dr. Naibedya Chattopadhyay. For this, I request the chairpersons to introduce the speaker.
You welcome, Dr. Naibedya. It's my pleasure to introduce and very interesting topic. Dr. Naibedya is Chief Scientist, CSIR, Central CDRI, Lucknow, and Assistant Professor of Medicine, Harvard Medical School, Boston, USA. I'm going to, as I was reading your topic, with combining the fat with the osteosarcopenia. So, as a physiologist, very exciting for me to listen to this. So he will be speaking on activating adiponectin receptor as a potential pharmacotherapy for mitigating osteosarcopenia. Dr. Naibedya, please.
Good afternoon. At the outset, I would like to express my sincere gratitude to the organizers for giving me the opportunity to share my data. This is the capacity in which I have been for the last 4 months. The research which I'm going to share with you have been done while I was at Central Drug Research Institute. And this is going to be my conflict of interest declaration arisen from my tenure at Central Drug Research Institute. So very briefly, what you come to know from once I finish my talk, some intimate relationship, the nature of the relationship between bone and muscle, and then adipocyte will kick in to this interaction, and why this interaction between bone and muscle is important, and then I will touch upon our attempts to discover peptides that can serve as adiponectin receptor agonist as well as small molecules. So if anybody is interested after my talk to know more about osteoporosis and sarcopenia, which coexist most of the time as the recent data is revealing, and together this terminology they use is osteosarcopenia, one might be interested in reading the special issue that has come out in Journal of Endocrinology and Journal of Molecular Endocrinology, which I have edited. So it is a common sense these days that exercise builds not only bone but also muscle. And weak muscle is synonymous to weak bone and vice versa. So what are there's some problem with this slide. So that is why we say that osteoporosis often coexists with sarcopenia. Now, if you see the crosstalk between bone and muscle, from the muscle side, there are factors that are secreted, which include IL-6, myostatin, and irisin. Of these, IL-6 is a villain. So its increased production would lead to loss of bone, whereas myostatin and irisin, particularly irisin, which is produced when you are exercising. The exercising muscle produces irisin. Its other name is FNDC5. So myostatin and irisin, they are produced as a muscle anabolic, but at the same time, they increase BMD and osteoblast differentiation and promote bone formation. And from the bone side, which is called, so these were so-called myokines, and these are the osteokines. And of these osteokines, osteocalcin, osteoprotegerin, and insulin-like growth factor, these, when they're produced by bone, they help in muscle mass building. Whereas when there is bone loss condition, such as osteoporosis, these two things go up from the bone, rank ligand and sclerostin, and they not only are detrimental to the bone itself, but also it inhibits myogenic differentiation and is harmful to muscle. Now, fat is coming into the picture because I'm introducing adiponectin. Now, adiponectin is predominantly produced by white adipose tissue. And adiponectin is the most abundant adipokine in the body. In the circulation, adiponectin is at the level of the range anywhere between 5 to 30 microgram per ml. So that is the range of adiponectin in circulation. And it is produced predominantly by white adipose tissue. But later on, it was discovered that muscles also produce, skeletal muscle also produce adiponectin. So looks like adiponectin could be a good connection between muscle and bone and acting both on muscle as well as bone. Now, what is adiponectin? Adiponectin is people call it a cytokine, I call it adipokine, and it has multiple domains, the signal peptide domain, and then there is a variable domain, and then there is a collagen domain. This is a collagen domain. And this collagen domain actually imparts the ability on adiponectin to oligomerize. Like all those oligomerizing proteins like collagen, laminin, TNF-alpha, they have certain signals or amino acid peculiarities that enable them to, about which I will come a little later, which allow them to oligomerize. And adiponectin is present in circulation never as monomer, always in multiple oligomer format. And then there is a globular domain which is present in circulation once it is cleaved enzymatically. A globular adiponectin is there, and globular adiponectin, GAD, circulates as trimer. And adiponectin as a whole can be present in the bone, in the serum as low molecular weight trimer or medium molecular weight trimer. That means when two trimers are bound is a medium molecular weight, and then if more than two trimers are bound together, it is high molecular weight adiponectin oligomer. Now, adiponectin receptors are reverse or I would say inverted G protein coupled receptors. So that extracellular domain is the C terminal is the extracellular domain, carboxy terminal end, and the amino is amino terminal amino site is intracellular. And there are two forms mainly R1 and R2. And this signaling is triggered primarily physiological circumstance by high molecular weight adiponectin, primarily by high molecular weight, also to some extent by globular adiponectin. So now you can imagine that you cannot use this protein in any form in a therapeutic mode. Now about the signaling, how does it signal? Well, it is an inverted G protein coupled receptor. And for any surface receptor, you have to have adapter protein. In this case, it is APPL1. And its primary target, or it has a fast acting, means rapid acting component, and a slow acting component. The fast acting component is induced by AMPK. AMP, you know, the nutrient sensor of cell, and it rapidly phosphorylates AMP and leads to ACC phosphorylation, induction of PGC1 alpha, which is a mitochondrial biogenesis factor, and together they promote fatty acid oxidation, mitochondrial biogenesis, and then with the help of P38, it induces glucose translocation, GLUT4 translocation for glucose uptake by particularly muscle, induction of AKT leading to increased glycolysis. It also has NF-kappa B mitigating effect. That means it is anti-inflammatory component is quite pronounced. And then there is a slow acting component, which is PPR alpha. When it is induced, then it leads to fatty acid oxidation as well as decreased fatty acid synthesis. So overall take home message is that it will promote fatty acid oxidation, utilization of fat as an energy source, improve glucose uptake, and inhibition of gluconeogenesis. So what are the impacts of adiponectin in the metabolic disease? First of all, estrogen is something which induces adiponectin production. And adiponectin itself induces its own receptor. So there is a vicious cycle positive feedback loop operating here. Now, there is a system level metabolic integrator. I alluded to the fast signaling versus delayed signaling. So adiponectin receptor R1 type actually is responsible for the fast signaling by AMPK phosphorylation, whereas the slow signaling is induced by zinc domain, which imparts in the transmembrane domain, it imparts ceramidase activity in adiponectin receptor 2 that induces the breakdown of ceramide to S1P, sphingosine 1 phosphate, leading to induction of PPR alpha gene transcription. Okay, so the linking is that fat and bone shares a common origin. That means mesenchymal stem cell. In the morning, you have heard about one lecture where the mesenchymal stem cell being is a common origin for bone, for both osteoblast as well as adipocytes. Adiponectin levels are inversely related to BMD and postmenopausal osteoporosis. So these are the linkages we looked into before coming to addressing the role of adiponectin signaling in bone and muscle. Muscle, as I already said, is a major source of adiponectin and express adiponectin receptor R1, and bone cells express both type of adiponectin receptors. Now, before I come to the relationship between the effect of adiponectin and bone, let me very briefly tell you about the backbone of bone biology. In all our adult skeleton, all our board, all the audience people sitting here, at least at 1 million sites in weight-bearing bone, this remodeling cycle is going on. There is an activation phase, and the activation phase is when the osteocytes, which are the major type of bone cells, more than 95% bone cells is osteocyte. They're connected to each other like neurons. But when there is a micro damage, when the osteocyte connection, the canalicular connection is broken, those distressed or impaired osteocytes, they send a distress signal which is rank ligand. And the rank ligand is the most potent osteoclastogenic cytokine that will lead to production of formation of osteoclast that will remove the damaged bone here. This damaged bone will be removed by osteoclast. And then when they're removing the damaged bone, the bone matrix has a bound bone morphogenic protein, which is released from that bone matrix that will induce osteoblasts to come to the site of resorption called reversal phase, and then there is a lengthy process of bone formation done by osteoblast. Generally speaking, under normal remodeling stage, the resorption amount is equivalent to the formation amount. But when the postmenopausal osteoporosis or any kind of osteoporosis condition, particularly not only postmenopausal but as well as secondary osteoporosis caused by most commonly by glucocorticoid, in that case the resorption far exceeds formation. The drugs that are used are anti-resorptives, which are bisphosphonate or rank ligand antibody at this stage, and the formation drugs are teriparatide and abaloparatide at this stage. Very recently, romosozumab has received approval, but I'm not going to talk about it here. So here this is. So these are the two markers of bone formation and resorption, means bone remodeling or bone turnover markers, which assess to some extent accurately the status of bone remodeling. So PINP is the formation marker. It is produced by osteoblast, and CTX is formed by the action of osteoclast. This is a resorption marker. Under normal circumstance, these two markers will balance each other, but under osteoporotic condition, the resorption marker will be higher than the formation marker. So just keep this in mind as I will allude to this kind of concept while sharing my data. Now, the first question that we asked is that, does adiponectin has any direct role in bone? So to do that, I said that we can't use high molecular weight adiponectin because it cannot be therapeutically used the way it circulates in the blood. But globular adiponectin can be used, but with the caveat that it acts only on adiponectin receptor 1. So we have a globular adiponectin. Dr. Shobhoshaji Sannal in CDRI, he used recombinant technology to produce large amount of globular adiponectin and shared it with us. And here what we found is that this is osteoblast differentiation assay, and globular ALP is produced when osteoblast is differentiated, and in vitro incubation of globular adiponectin induced ALP. And then when these osteoblasts were treated with 10 microgram per ml of globular adiponectin, we saw a rapid induction of phospho-AMPK, which is the rapid signaling. I said that ADIPO-R1 has that feature, and globular adiponectin is doing that. And then we found that adiponectin receptor R1 is induced, and as I said, that there is a positive feedback, and ADIPO-R1 level is going up at 72 hours. And then PGC1 alpha, which is a mitochondrial biogenesis factor, is also going up by 36 and 48 hours very clearly. And when we added a dorsomorphin, which is a phospho-AMPK inhibitor, we saw that globular adiponectin induced PGC1 alpha induction was completely mitigated. And when we did that experiment in a molecular way. That was a pharmacological approach. This is a molecular approach using siRNA. We found the same answer. And then we found that when we did the extracellular flux analysis, we found that oxygen consumption rate was significantly increased, almost doubled by presence of globular adiponectin, and PGC1 alpha knockdown completely abolished the effect. And then when we used rotenone, which is the TCA cycle, the electron transport chain inhibitor, then the effect was abolished in terms of differentiation, but not by deoxyglucose, suggesting that only oxidative phosphorylation drives osteoblast differentiation by adiponectin, and glycolysis component is not required for adiponectin derived osteoblast differentiation. Then what we found was that in overectomized animals, which is an osteoporosis model available preclinically, globular adiponectin at 100 microgram significantly restored, and this is a therapeutic. That means we first let the animals lose bone, and then we started their therapy compared with PTH as well as alendronate, which is bisphosphonate. And we found that adiponectin completely restored volumetric BMD. PTH was better than globular adiponectin, but one must remember that globular adiponectin animals had 17% less body weight than PTH treated animal, and mechanical loading contributes to BMD. So that component, there is 17% less mechanical loading due to less weight by these animals. So if you factor that in, then it is equivalent to PTH. But this data, what I am showing, is not normalized to body weight. And then we found that the bone strength was also completely restored by globular adiponectin. This is a surrogate of fracture. That is why it is important to measure bone strength. And this is a dynamic histomorphometry to suggest the bone formation, to assess bone formation rate. That is the gold standard for measuring bone formation. Is not P1NP, what I showed, is a surface referent bone formation, double leveling of calcium. You see these double leveled surfaces. These double leveled surfaces are which are present here but not in the alendronate panel. And so bone formation rate was almost equivalent to approaching to that of PTH. And then in their bone, what we found was that ADIPO-R1 level, PGC1 alpha were both reduced in overectomized animals, whereas globular adiponectin treated animal had their levels equivalent to that of sham control, over intact control. So not only the primary osteoporosis, which is postmenopausal or over-induced osteoporosis, we then tested, does it have any effect on sarcopenia? Now, what we found here is that the overectomized animals, this is a gastrocnemius muscle histology of just H&E, they have angular and shorter fibers, and that was significantly improved with globular adiponectin. So cross-sectional area as well as ferrets diameters were improved by globular adiponectin, not completely to the sham level, unlike bone, but to a significant extent. And these are the assessment of the myogenic proteins, or one is the catabolic protein and myogenic protein. MyoD is the myogenic protein that was reduced in overectomized animals, that was restored by globular adiponectin, whereas atrazine, MARF1, these are catabolic protein that were increased in OVX, was reduced by globular adiponectin. ADIPO-R1 and PGC1 alpha component completely mimicked what was found in bone. So this is what our proof of concept was obtained, but again, globular adiponectin is not a viable option for clinical translation. So we wanted to see if we can have a short peptide derived from the collagen domain. Why collagen domain?
This we did in collaboration with our peptide chemist in CDRI, Jimut Ghosh. And what he designed is a short peptide of 12 mar, 14 mar, I think, 14 mar peptide from the collagen domain. And it has glycine and histidine repeat. And why he did that? Because with that, they can make oligomer. And with that oligomerization, they can higher level of structure should be obtained. And with that, he first showed that in comparison to insulin, in L6 myocyte, two D-glucose uptake studies in vitro was done. And he found that whereas in 20-minute insulin showed a sharp glucose uptake by L6 myocyte, in case of ADP1, this peptide, there is a sluggish increase in glucose uptake. So that is the difference between adiponectin-induced glucose uptake versus insulin-induced glucose uptake. And then he found that when metformin compared to metformin, the phospho-MPK, metformin, the anti-diabetic drug, the first-line anti-diabetic drug, and the peptide drug, both of them induced phospho-AMPK in L6 myocyte. And then when the animals were treated, DBDB animals were treated with this ADP1 peptide, by 15th to 20th day, you get a clearly by 20th day, you get a nice increase in insulin, serum insulin level, and glucose utilization, OGTT was done, was significantly increased, improved by ADP1. So we used that one and found that in overectomized animal model of osteoporosis, it clearly induced nice restoration of BMD as well as in both L5 lumbar vertebrae as well as femur, these two weight-bearing bones that are prone to fracture. And then PINP, the osteogenic marker, was restored, as well as this CTX1, the resorption marker in the serum, was also restored, which goes up. The resorption goes up, which was restored. So this is what we call it rebalancing bone remodeling. So you have seen this, I have introduced you to this cartoon. So in this case, what happens is that we started with animals where the bone formation component was higher, CTX was higher than PINP, and we were able to, using this ADP1, we were able to bring it to equal level. So this is called rebalancing bone remodeling. Now, as Jimut Ghosh was trying to make a new product patent out of that peptide stretch, because that peptide stretch cannot be patented because it is an endogenous peptide. So nature has created that peptide. You can't get patent on that. So you cannot develop it in any way like a drug fashion. So as he was trying to make modification in the peptide to obtain product patent, I and Shobhashaji Sannal started wondering about how about the small molecule targeting adiponectin receptor. And then we started literature search with three keywords: obesity, insulin resistance, and phospho-MPK. And small molecule, actually, 4. And using these, we found that the common theme was coming in terms of the chemical scaffold is quercetin or isoquercetin. This is the compound that has the capability to induce rapid phospho-MPK, insulin sensitization effect, and anti-obesity effect in preclinical setting. And then actually I was not surprised because I already have worked with both camphorol and particularly quercetin. And I found that quercetin in two different circumstances has bone anabolic effect. But very interestingly, we also have found that this compound, which is a quercetin analog with C-glucoside, which is a very uncommon kind of a natural compound. The C-glucoside ring, covalent linked, glucose is covalently linked with the flavone ring. And that one we also did a detailed study. So this is the compound, and this turned out to be a novel compound, means natural compound but novel compound, and this we have US patent on it. And this compound, when given to osteopenic animals at 1 milligram per kg, it was as good as, in terms of restoration of bone volume in osteopenic animal, it was as good as PTH teriparatide. And not only in these overectomized animal, even in glucocorticoid induced bone loss, it restored when we gave it with methylprednisone, GTDF and PTH, they were equivalent in their activity. So then we studied what is the mode of action of GTDF, and that is when we took help of Professor Godbole. He was also my PhD mentor, believe it or not, sitting there. With his help, he radio labeled GTDF, and we did this receptor binding assay in HEK cells transfected with adiponectin receptor R1 and R2, and chased with, sorry, not HEK, it is CHO cells, and was chased with cold GTDF. And then we found that this compound GTDF has adiponectin receptor specificity almost 70-fold higher than adiponectin. R1 specificity is 70-fold more than R2 specificity. And it is, being a natural compound, we always are skeptical that it most probably is binding to 100 different things. So because of inverted GPCR structure of adiponectin receptor, and having found by this radioreceptor assay that it has selectivity for AdipoR1 over AdipoR2, which is quite a vast selectivity, 70-fold is not a small selectivity. Then we did a GPCR profiling, and 158 GPCRs were screened, and none of them have any binding. GTDF had no binding with any of these 158 GPCRs. So which suggests that it is a very specific AdipoR1 agonist. And then when we used in the C2C12 myocyte, the the adiponectin receptor downstream signalings like phospho-MPK, ACC, phospho-ACC, phospho-P38, and AdipoR1 and R2, in all cases, the induction done by GTDF was abolished by AdipoR1 siRNA. GTDF improves metabolic parameters in DBDB mice. IPGTT, intraperitoneal glucose tolerance test was done, and it significantly improved compared to the vehicle-treated animals. And HbA1c level in 28-day was significantly reduced in these DBDB animals. Now, what was very impressive was when we studied their pancreas. So this is insulin staining, immunostaining of insulin. So if you see that the vehicle have quite a damaged islet, even at 40x, you don't see much insulin staining, but when you treat them with GTDF, you see nice insulin staining of islet cell here. And also, the apoptotic cells which are present in the vehicle animals, which is the pink one, Ki67 and tunnel staining were done together. And you see that the apoptotic cells are also significantly reduced. The tunnel positive cells were significantly reduced. And then another important thing happened, that the animals achieved the capacity to utilize the energy much more efficiently. How? They converted their white fat into brown fat, as revealed by UCP1, which is a marker for brown fat. And you can see that GTDF has significantly strong, significantly strong UCP1 staining. And remember that the brown fat typical if you do H&E staining, you will see that the fat cell, that the cells have shrunk. So typically brown fat characteristic it has taken. So what happened here? That the restores glucose utilization, restores beta cell health, and induces browning of fat. Three things has happened. And then when we looked at their bone, DBDB bone, what we found was that it was significantly reduced BVTV in case of DBDB animals with GTDF and metformin. There was significantly enhanced bone formation. And PGC1 alpha levels which were reduced but induced by GTDF as well as metformin in the bone, and MARF1, the catabolic protein, was significantly increased in the bone of DBDB animals, but in case of GTDF and metformin, it was reduced. So what happens in summary, that GTDF not only improves the metabolic health, but also enhances bone mass with attendant increase in PGC1 alpha and decrease in MARF1 level. Not only that, it protects skeletal muscle against skeletal muscle atrophy, and this was done with dexamethasone treated animals, and you see that the fiber levels, myofiber levels are significantly shrunk in case of dexamethasone, whereas these are restored. In this case, the myotube area was plotted, and this is the immunohistochemistry. So ferret's diameter and cross-sectional thickness all were restored by globular adiponectin when it was treated to animals that received dexamethasone. Remember, dexamethasone is a synthetic glucocorticoid which is very harsh on animal. Animals become lean within 15 days, they lose muscle, they severely lose bone, and if you continue for another week, they will die. So in these animals, to really improve muscle and bone is something very strong effect. And this is PGC1 alpha, atrazine, MARF1, and MyoD, the same way we studied in case of overectomized animal. A similar trend was obtained that biogenesis, mitochondrial biogenesis was enhanced, and catabolic proteins were also was decreased. Now the problem was that with two stereo centers, this GTDF, we licensed this patent to Chemistry LLC, a New Jersey company, and with two stereo centers, they took the patent and they put their smartest of the chemists, and they found that if they are able to synthesize it, it will cost a fortune. So it is not industrially scalable. So finally, we started to look at a simpler compound having a similar effect like GTDF. And there we hit upon this apigenin, on which also I worked when I was in Boston. Also, I worked on apigenin. And then we found, to cut the long story short, when we did a series of screen of apigenin related compounds in vitro using ALP assay in osteoblast differentiation, we found that isovitexin, apigenin C glucoside, behaves like adiponectin receptor modulator. This is the first example. This is isovitexin. This is the structure of isovitexin, apigenin C glucoside. Phospho-MPK is induced in 15 minutes and goes up to 45 minutes. Apigenin is not able to induce that rapidly. That shows the selectivity posed just by this sugar group. And ALP activity, which is for osteoblast in differentiation, that was induced, was reduced by or mitigated or abolished by SI-ADPO-R1 for isovitexin. It induces oxidative phosphorylation as assessed by seahorse, about which I have shown some data before with globular adiponectin, remember. So in this case, we employed the same experiment, and ATP production, which was strongly induced by isovitexin, was completely abolished by SI-ADPO-R1 as well as R2, which suggests that this is actually a dual agonist. And in this case also, rotenone was able to completely mitigate osteoblast differentiation induced by isovitexin and not to deoxyglucose, similar effect as globular adiponectin, which suggests that again, in this case, like globular adiponectin, isobutexin is fueling osteoblast differentiation by oxidative phosphorylation. Now, isobutexin has a much better oral bioavailability than apigenin. So oral bioavailability of apigenin was only 4.6%, whereas in case of isobutexin, it was close to 15%, which is quite impressive when you reach a double-digit oral bioavailability. So we are now, CDRI is now working on new analog for isobutexin. But in the meantime, we tested their effect on overectomized animals, and it was very similar to the BMD restoration by isobutexin was very similar to PTH or teriparatide, and PINP level was restored and CTX level was reduced, so suggesting like ADP1 that it is able to restore the bone remodeling balance. So in conclusion, glucocorticoid mitigates osteosarcopenia. ADP1, a 13-mer oligomeric peptide which can assume oligomeric structure, can mitigate osteopenia. GTDF, a novel small molecule orally active ADPO-R1 agonist, which is a selectivity for ADPO-R1 and mitigates osteosarcopenia. Isobutexin, an ADPO-R agonist, mitigates osteopenia, and it also mitigates sarcopenia. I have not presented the data because my PhD student Chirag Kulkarni, who is sitting here, who has presented it in parallel symposium. So, but to include his finding, the last conclusion would be isobutexin, an ADPO-R agonist, mitigates osteopenia as well as sarcopenia. So future perspective would be to design new analogs of isobutexin. So it took us almost 14 years to come to find a possible scaffold around which chemistry can be done, new chemistry, medicinal chemistry can be done, and design new composition in terms of peptide ADP1, which is a 13-mer oligomeric peptide. So I must acknowledge my fellows, of which only Chirag Kulkarni is sitting here. And in terms of CDRI collaborators, I'm particularly indebted to all four of them, Shobbosati Sannal, Jia Urgain, Jimud Ghosh, and Rakesh Maurya is the medicinal chemist who discovered GTDF and isobutexin. I also have outside collaborators from Dr. Naveen Kumar, whose data I have not shared, who contributed in bone quality assessment. But another collaborator, come my PhD mentor, Professor Madan Godbole, is sitting here, whose data I have shared with you, and my lab members as well as funding from CDR, from various funding agencies. Thank you so much.
Vaidya, thank you for sharing 15 years of your journey. And I see this adiponectin receptor is quite promiscuous. It seems to be bound by the peptides and also by the chirols. Very interesting. And also seems to be the sort of all the, perhaps the variety of things, just apart from adiponectin, could you also find other chemicals or peptides in the body which bind to adiponectin, or is only one adiponectin, the adiponectin receptor?
Oh, endogenous?
Endogenous.
Endogenously, no, it is very selective for its, as such, they have multiple forms, as I said. Low molecular weight, medium molecular weight, high molecular weight oligomers, they will bind to it, as well as globular adiponectin, which is cleaved from. Other than that, endogenously, no other protein is known to bind to adiponectin. That much is pretty clear about it.
So, but very interesting that there is something which is a peptide and peptide binder seems to be binding to sterols and quercetins and everything.
Yes, true. And it is, as you said, it is promiscuous, means from brain cells to everywhere it is there, and we are only able to understand their metabolic part, function of it. Because adiponectin level So its level will go down in obese patient, its level will go down in insulin resistance. So as these days you are hearing so much of noise about GLP-1 agonist making news. Adiponectin, the problem is that it is very difficult to drug that receptor.
Okay.
So that is the reason why it is not being really targeted. I'm sure people are working on it as we worked on it, but the correlation is very clear. You wouldn't find a single person, lot of data are available, meta-analysis available, that obese patient has less adiponectin level and type 2 diabetes patient has less adiponectin level.
And what about those who are because of this?
And another thing, females have more adiponectin than male.
Yeah, I was coming to that.
Yeah.
So now there's this trend of being shredded. If the people want to have a very thin physique and lose fat completely. So in that case, what is your understanding of how it will impact the bone growth? And everybody wants to be very thin.
Thin people, yeah, I mean, thinness, I mean, if you go by Dr. Yajnik's theory, particularly for Indians, he says that thin fat. So we have thin fat. So our thinness is actually of not any health, but it is to our disadvantage that obese people have actually more bone mass because they have slightly more weight. And
Correct.
reduce in that score as well as in terms of metabolic health or metabolic fitness, we are not very fit. So adiponectin level in terms of BMI not yet been studied to that meticulous extent compared to obese people versus non-obese. But BMI's gradation-wise adiponectin data is
No,
not.
in this regard, there's one study going on, getting place for 3 decades. So they're tracking people from the age of 70, 70s, 80s, and 90s. They're the 90s. The only factor which matched with longevity was a little bit of fat in them.
A little bit of fat in?
So a little bit of increased fat was correlated with, can't say caused it, but correlated with longevity.
Oh, longevity.
Yeah. So across all the things, whether they do vitamins, whether they do exercise, whatever, one of the consistent factor was that. I mean, and with this, your knowledge which you're giving, perhaps the combination of the whole thing, that some amount of fat is necessary as a source of adiponectin and adiponectin receptors, and then glucose metabolism, everything seems to be linked.
Yeah, I mean, in terms of fat metabolism, what adiponectin or in terms of type of adipose tissue, as when we saw that data, our data, that adiponectin level will be more beneficial in terms of switching the fat type from white to brown. That is what we think.
Are there any queries from the house? No.
Adiponectin is more in females. Does it have any relevance to where they have a pre-menopausal to post-menopausal? The time of duration can vary from 1 to 5 years. Is there any circulating levels studies known in these 5 years?
Yeah, when I said more in female means female in their reproductive age, and it is well demonstrated, not by us, but by many others, that not only estrogen, but any estrogenic compound will stimulate adiponectin production as well as its receptor. So women, till the reproductive age, they have less metabolic problem compared to male, for which attribute is given to adiponectin signaling. But after menopause, preclinical data we have seen, I have shown data in bone that adiponectin receptor was down in the bone of overectomized animal, clearly showing that it is estrogen dependent. So you can easily extrapolate to clinical condition, human condition, that after menopause, their adiponectin receptor level is low, and they are not getting much favor from. So that is the reason they're gaining weight. Remember, they're gaining weight.
Cushion for the transition.
Transition, yeah. So in terms of epidemiological data wise, I can't really say how clean is the data, But in animal data, it is very clear that the moment you remove estrogen source, adiponectin signaling would be affected. And it starts from the receptor expression, downregulation of receptor expression.
Thank you, Dr. Naivedya,
Thank
for
you,
your time and exposition.
Yeah.
Now I request Dr. Ashok Jarial sir to present a certificate of appreciation to our speaker.
Give me a second, please.
Thank you, sir.